Thank you. It's great to be out here at front today. I should say it's morning. It feels like the night though, doesn't it? So if those people can come and get a seat, if you can, that would be great. Although, you know, I can understand standing up as well. I'd love to do more of that myself. So welcome everybody to Research and Clinical Excellence Day. Thank you so much for attending. We are really honoured by the speakers we have today. This includes trainees right through to clinicians. And so there's a broad breadth of research going on at UCSF, from the very, very basic research biology that's happening right now at the bench, I hope my people are up there either here or at the bench, and it's transitioned way through all the spectrum through to the other end of clinical application. And so you're going to hear about that today, so please be engaged in these talks. So we've got a very large number to go through, and please be here for these talks. I know there's other options to do today, but this one is your best option, I can guarantee you that. We've got a plethora of trainees who are going to give us an update on where they've been to with the journey through their projects. And of course it is a journey, I know it is a journey, from the beginnings of understanding research to going through just a 3-month program is enough for you to understand where you're at and where you're going to go in the future. That's obviously extremely important. Then we have a number of senior talks, and I'm actually going to give one too, and if you don't come, I'm going to be very upset and crying up here. No, I will be upset and I will come and get you. All right? So think about that, 2:45. Where are you? Love to see you. And then we'll obviously go through the whole spectrum today, and we're going to end up with the awards at the end. Now, I think there are raffles. Is that correct? Is there a raffle? Yeah? Okay. Who wants to win? I do, so I'm going to win. So you can't win, okay? But if you want to go against me, I'm also happy with that. I like a little competition, but this one's the winner right here every time. So, but we'll go through the day, and there will be prizes to be awarded. Please have these bottoms on these seats for those prizes, and then by 4 p.m. we should all be ready to go, and there'll be essentially a lovely drinks and essentially water. No, there'll be a lovely after evening session for you to join in if you like to as well. Okay, that's all from me, and I'm now going to introduce your Dean Reddy.
Well, thank you, Sarah. And welcome everyone. It's welcome to Research and Clinical Excellence Day. So I'm delighted to see so many of you here, to see this room full exploring science. You know, this is an enduring tradition that we are very proud of. Did you know this is our 20th year that we've held Research and Clinical Excellence Day? 20 years. Congratulations, everyone. And it is one of the days that I like most about the academic year. It's a day of sharing discovery. So my name is Mike. My pronouns are he and him. And, I have the privilege of serving as Dean of the School of Dentistry. So what's unique about Research and Clinical Excellence Day? It's a day that celebrates what is unique and special about UCSF School of Dentistry. And what is that? So it's the spirit of discovery that infuses our community and the incredible science that comes from that passion. It's the doctors and researchers who advance our clinical pursuits that lead to better outcomes for patients. And of course, it's our bright and ever-curious students who combine their mastery of science with compassionate care to make a difference in the community. Coming to this meeting, I was thinking about research, and I came across a research publication in a journal that probably not many of you read. It's the British Psychological Society, and they had taken a deep dive into looking at dad jokes, which is something personally close to me, right? And they found that even if they're a little cringy, right? And, in reality, we all know they're truly not funny, right? They have the capacity to make us better humans, And it turns out that dad jokes are actually good for you. So I need someone to explain that to my kids. So, okay, so here goes. The first years have heard some of these probably already, but why did the toothbrush cross the road? It wanted a change of paste. I told you they're not truly funny, Yeah. Yeah. All right, yeah. My dentist asked me if I floss between meals. No, usually between teeth. Yeah, well, all right. So why does this day matter so much? It's inspiring to see and hear from such an amazing, talented group of people. The learners who have been working so hard to become future leaders in dentistry and health science. The dedicated faculty who serve as mentors and coaches. And the experts whose lectures add to our knowledge. But I think the real power of this meeting and of all research meetings is the cross-communication and conversations that happen in between the posters and in between talks, the exchange of different ideas that occurs. A few years ago, I was invited to give a talk at the American College of Rheumatology. So a couple of things surprised me. First, why did they invite me to speak? The second was that the meeting was so large. There were thousands of people at this meeting. I didn't realize it was that big. So I started off and was giving my talk, and I was really there to talk about some imaging to go ahead and measure small amounts of bone loss. But I started off by talking about biofilm dysbiosis leading to an altered immune response, and that inflammatory host response leading to the destruction of bone that we see around teeth as periodontal disease. In the middle of my talk, someone got up and asked a question, and it went something like this. So you have this complex microbiome host-modulated disease. Why do you insist on treating it with a piece of string? When the laughter died down from the crowd, I told requested the question and said, well, the second half of my talk is about measuring small amounts of bone loss and the potential of host-modulating therapies that are commonly used in arthritis to manage disease. And it turned out that after we talked afterward, and we ended up working together and actually writing a paper. So my advice to our learners today isn't to throw away the floss, but it's to ask what you may think is an obvious question. It could lead to a productive new approach in our research. Today we're honored to have a very special guest join us, Dr. Saul Villeda, who is in the Department of Anatomy here at UCSF. Everyone's told me just a brilliant researcher and a terrific speaker, so I'm looking forward to, and we're going to introduce him in just a minute. But I also want to recognize our other featured speakers who we'll be hearing from today. Dr. Perry Vallupillai, who is an oral medicine clinician at UCSF, will be presenting our spotlight on clinical excellence. And Dr. Sarah Knox, our Assistant Dean of Research, is the recipient of the Faculty Lecture Award. Congratulations. She's going to share her extensive research into salivary glands and how she's been working on bringing her discoveries out of the lab and into the impact of patient care. So these are going to be some illuminating talks. Thank you, each of you, for contributing today. I want to give a special thanks to the organizing committee for putting this together. You've done a great job again. Thank you. And the John Green Society for producing this unique event. And now I'm going to go, wait, that's not, sorry, my apologies. I'm going to turn things back over to Sarah to introduce Saul.
Okay, it's such a delight for me to introduce you today to a great friend of mine, Dr. Saul Villeda. So Dr. Villeda is a current associate professor here in the Department of Anatomy. He serves as an associate director for essentially the Bakar Aging Research Institute, which we like to call the Barry. He obtained his Bachelor of Science degree at UCLA, so that institution you might know down south. And then he went on to get his PhD in neuroscience at Stanford. Of course, then he came here and he started his independent research career as a Sandler Fellow. So Dr. Villeda has made some amazing discoveries, and the ones that I truly love and what he's been investigating wholeheartedly is the ageing process in the brain. And you know where the brain is, that's right, it's right next to the oral cavity. So he's looking at ageing processes and how they can be reversed through altering levels of circulating factors in the blood. So he's done a lot of research in this area, and it's been phenomenal. He's going to be talking about those phenomenal findings. I want to tell you about his, want to point out his accolades to date, and there are many, many of them, and here are some highlights. He got awarded the NIH Directors Independence Award, the Keck Foundation Medical Research Award, Glenn Award for Research in Biological Mechanisms of Aging, McKnight Innovator Award in Cognitive Aging, and recently here at UCSF, the Byers Award in Basic Science. So I'd like to introduce to you today Dr. Saul Villeda.
Hi, everyone. It's a pleasure to be here today, and I'd like to thank also Dr. Sarah Knox for the invitation. So I thought, okay, you're probably thinking, we're going to hear a little bit about cognition, the brain, how is that going to relate? And what I thought would be interesting, and what I talked to Sarah a little bit about, would be that I'm going to share my research and my science with you, but you're being exposed also to the researchers, right, and scientists. And oftentimes when you come and you hear a scientific talk, you get to see all the data, and you get to see the p-values and the hypotheses and the questions. We don't necessarily get to know the researcher as well. You don't really get to know who they are. And intimately associated, it's totally like connected to the reason that I do the research that I do, is who I am as a person. You know, me being a scientist is the same to me as who I am. It's part of my identity. And that association with my identity, how I perceive myself, is the reason that I went on to study aging. It's the reason that I went on to study cognition. So I thought as part of my science talk, I'd give you a little bit about my personal trajectory as well as the science and how that led. So hopefully, you know, at the end, I'm happy to take questions on any and all of it. And I hope that this also highlights how important it is to really carry who you are, your experiences into both either research as well as when you're seeing patients, because we're not all the same, and that's really going to influence what we do as well. So why do I have this image? So I'm going to talk to you a little bit, you know, scientifically about the brain and the blood. And in a nutshell, I love studying the brain by not even looking at the brain. I love studying the brain by studying outside of the brain, the body. And I really focus on that communication between what's outside and what's inside. And I'm fascinated by this idea of cognition, but not just the way we think about it. You know, when we think of cognition, oftentimes we think psychology, or we think immediately disease or dementia, and I will get to that. But what got me into the idea of cognition is really more about who we are. I'm really interested in this idea of identity, right? Every morning you go and you probably have a mirror. You're going to brush your teeth and you're going to see yourself, maybe wake up, and instantly there's that recognition, that association of who you are, that identity, the way you look, the way you feel, what you have to do that day, what you did yesterday, maybe the next exam, the experiment that you have going. And we're a collection of those memories, of those experiences, of that growing identity throughout time. And then we get to that aging component of it when we're elderly. That's when we start having things like dementia. We start having loss in memory. We start having loss of connections. But it's not just our brain. Our entire body starts really having these huge changes. We no longer have the care that we used to because maybe we don't remember to have it. And I want to figure out ways of how do we maintain our sense of self, our identity, for as long as we can. And we know we're in peak form when you guys, like right now, most of you are like doing amazing. Actually, cognitively, you get better until you're about in your early 30s. So you guys have a ways to go. You're getting better. I'm now in my 40s, so we're kind of coasting. I'm all right. And then a lot of the people that are near retirement, they're kind of swinging downwards. And we're going to leave it there, though. But I'm fascinated by, can we leverage biology to maintain ourselves, to maintain that sense of identity? And that's really my overall question. My interest is, can we preserve our identity, our sense of self, over time? And can we do that by countering aging itself? Can we fundamentally learn something from biology that we can leverage towards health, and not just brain health, but all health? Is there something we can learn about the aging process to target every single one of those hallmarks that we're seeing? Regenerative decline, cognitive decline. I mean, everything that we think about, right? Bone, brittle, muscle, injury, all of it is declining, our ability to sort of counter that process. And is there something fundamental that we can learn from biology to counteract that? And I'm going to focus predominantly on the brain and cognition because of this idea of identity. So then, who am I, right? I'm talking about identity and how this is like my driving force. So I'm going to tell you a little bit about my trajectory, both personally, scientifically, and then how that led to my research and some of the research that we're doing, a little bit published, mostly unpublished as well. So it's about half and half. Okay, so a little bit about my personal trajectory. So, Sarah just said, okay, he went to UCLA and to Stanford, he has this award and that award, and this is why he's at UCSF doing great science, and that is true. However, that's not necessarily who I am, right? Something led me to here. So I actually was, I was born in LA. I'm the first one born here. This is like a picture of me in like the early 90s. But we all have our own identity, right, and our trajectory. So I came from a very poor part of East LA. I grew up during when it was like a lot of the gang wars were going on. So that's where I'm from. Growing up, it was often, we knew how to get like duck to the ground when there was like those loud sounds. That's where I grew up. So this particular scientist has that as his personal background, which you're not going to see in my CV. So like I said, I was the first one born in California, so I'm a first generation American here. My parents over here, they're originally from Guatemala, so they actually came here illegally. So I was the first one born here, they came here illegally in the 70s. And what was interesting is I had an amazing upbringing from a cultural perspective. It was really rich with like who was there, my family, the food, the environment, the music. It was fantastic. Within my community, within sort of where I was born, but I was part of a greater community, right? Because I was the first one born in the United States. And something really interesting happened about that because at home it was all about Spanish and culture. And as soon as I left that little neighborhood, everything was in English and I didn't quite understand it. And I knew it and I could understand what they were saying, but I was a little bit scared because it wasn't what I was used to. So that's the first time I kind of understood this idea of the imposter syndrome, which is I could engage with them, and I was good enough to engage with them, but I chose not to because I just didn't feel comfortable. It was foreign to me. And what really helped me, and really I think is the reason that I started going towards science eventually, I just didn't know that, was actually education. That was sort of the great equalizer to me. So I actually got really comfortable speaking English through programs like Sesame Street on television. So I did a lot of television because my parents worked and sometimes I got left alone. And PBS and like Big Bird were amazing. So this is predominantly how I was practicing sort of that language with Sesame Street. And they looked like me. There was a little bit of everything Sesame Street, right? That street is full of like, everything from green puppets to people from, the Caribbean. So I could see myself as part of that. And then that really helped me when I went on to public school. I'm a product of the LA Unified School District, and that's where I was really sort of exposed to things like mathematics for the very first time. And you know how I mentioned I was a little bit of an imposter syndrome? I had a little bit of a hard time engaging like verbally with people. Guess what? Math is silent, right? Math is, it's functions. I don't have to say a thing. I just write it down on a piece of paper. Awesome. So I really started liking things like mathematics. And mathematics opens the door to things like biology, to things like chemistry, to things like biochemistry, right? So that was sort of the introduction into a much bigger world of science. And it's funny because when I was growing up, I knew lawyer, and we all knew lawyer. They were like the most important people in our community was lawyers because we went to them to help. The next one was physician, right, doctor, because obviously you need medical care. And then the third one was engineer. I don't know how the engineer one got in there, but those were the three that we knew. However, when I started going into education, I realized like mathematics, science, they're not isolated. They're actually connected to health. Like the doctor, the physician that is treating me, the dentist that is making sure that like the wisdom teeth are coming out. They had to go through mathematics. They had to go through science. The engineer did as well. So I started realizing that it was more than just an individual, one class that I'm taking. It's part of a whole kind of idea and notion that we can leverage fundamental knowledge, leverage it, build upon it, use it for health, and then that can actually impact patients. And that is what got me really excited. And that's the reason I ended up going to college. That was a whole trip. If you want to find me afterwards, how on earth I ended up going to college, but I did. I went to UCLA. The reason why is because I could take, let's see if I remember this. I could walk to the metro station, take the train down to the central station, from the central station take 2 buses, from the 2 buses get to the actual dorms. And in my parents' mind, that meant I was coming home every single weekend. And I promised them that I would, and I absolutely did not. But that is why I ended up going there, which was great. Now, at UCLA, something interesting happened. I changed my major 13 times. I had no idea what I wanted to do. But eventually, I landed back on science, and I took this developmental biology class. And I learned about this experiment, which blew my mind. These are newts, and newts can be either white or black. And this actually won the Nobel Prize, a graduate student. And this is called the Spearman organizer. And what they did is they took these little embryos and they took a little piece of one colored embryo and stuck it onto the other one. And they just let it grow. And it turns out that you can actually form kind of a Siamese twin. But the Siamese twin is actually predominantly still from the host twin. So something about that little chunk, that little tiny chunk of embryo that you grafted onto the other embryo gave information to start making an entire new axis of an organism. So think about it. There's information in this little tiny tip that told the entire body, make a second one. And that blew my mind, completely blew my mind. That means that there's intimate communication signals that can make an entire organism appear. And that went on, like I said, to be called the Spearman organizer. But to me, it was still this idea of communication. I'm fascinated, right, by how we tell things to work and how we can engage with each other. And here I have an example of a tissue engaging with a whole organism. So I thought this is fantastic. And that's what led me down the road of biology. I'm like, okay, I want to figure out how things talk to each other and how you can manage to have this type of change, right? A few cells making a whole organism. I went and I actually then engaged in research. There's these amazing programs like the MARC program. There's a whole bunch of other ones at the undergraduate level that really promote kind of diversity and inclusion. And they also paid for my tuition, which was amazing. So I actually did research and I studied development, but this time of the spinal cord. And this is where pain is processed, and the nerves kind of innervate onto it. So I was really fascinated by, well, what are the signals coming from one part of the spinal cord that position those cells to be able to process things like pain, right? Because pain can be good, it can be bad, but it also can be good. So I was really fascinated by that. As part of that program, I actually got to come here to UCSF as an undergrad to do research. I did summer research. You'll often see these summer students coming from different universities. I was one of them. I actually did research with Dr. Alan Bosbaum, who now is my department chair. So you never know where you're going to end up, okay? So be nice to everybody. Make sure you do good work with everybody. So yeah, so he's now my department chair. And from there, I then ended up doing my PhD, like Sarah said, and I went on to Stanford. And at Stanford, I continued with this idea of development and this idea of communication, but in a different setting. I actually switched over to aging. Now, why? It turns out that at the time that I got to graduate school, before we thought stem cells are only in development. And then kind of biology got broken, and people were like, oh my goodness, stem cells are in the adult as well. And that was contentious back and forth, but yes, we have stem cells as adults. So that's pockets of development are left in your adult body. But what about aging? It turns out one thing that declines is regeneration, and you're going to hear work by Sarah that's looking at the salivary gland, but aging is like a radiation. It's going to completely destroy regeneration, oftentimes at the level of the stem cells. So it's could I then study development, but not in development? Could I actually study these pockets of development in aging and try and figure out not how to make a second organism form, but rather, can we kind of hack into that development and improve function in the elderly, in an old individual? But remember I said I really like this idea of communication. I love talking. So I wanted to know not just what are the cells that are there and how do we increase regeneration. I wanted to know what's the communication going on. Now, when I say aging, right, it's going to mean something different to all of us. I told you I'm fascinated by identity and cognition, so I think about the brain. You're all going to be thinking probably more about mechanics, or you might be thinking more about regeneration, or you might be thinking more about, is something sort of tearing or breaking down, or maybe the matrix, the matrix and how scaffolds are formed. All of that changes with aging, because aging is a whole body process. It's not just one tissue. So I started thinking, well, can I learn then something by looking at the whole body that could inform me about the brain? And I started thinking, what connects all of the tissues together? Similar to that little piece of embryo that signaled the whole other embryo, what is it that connects our body together? And the answer to that is really blood. Blood is connecting us all together. Every single tissue in our body is connected to blood. So then I thought, could there be signals in our blood that are either promoting aging or that are promoting function or youth? And in order to do that, you have to manipulate all of blood itself. So I made use of this crazy old surgical technique. It was first described, I believe, in the late 1700s. There are some crazy papers from like the 1920s where they only have like sketch drawings on them. They sewed everything you can think of together in like the early 1900s. I limit it to a young animal and an old animal, and then we connect them together. It's literally a surgery. You basically make Siamese twins. And then you can ask two questions, right? You have this young animal receiving the blood circulation of the old, and then you have this old animal receiving the blood circulation of the young. Now, during my time at my PhD at Stanford, what we discovered was that there are active aging signals. As you get older, your body is literally telling your body to get old. So there's some reason for it, and we can get into the evolution and the theories, and well, don't you want to get rid of the older people because the younger people need food, and then we can go back and forth on it. We don't really know why we age, but we do know that we age, and we think that part of it is an active process. It's not just wear and tear. It's not just like, okay, because you've been around for a long time. There are some signals that are actually promoting that, and that's what I discovered when I was at Stanford. That's the reason that I ended up getting sort of my job here. As part of like my scientific trajectory, though, I will say that it also influenced my personal trajectory. That's also, I met another scientist. He's my husband here, Greg. You might see him around campus as well. We were both fascinated by this concept of kind of aging, neurodegeneration, and he's actually been on almost every single paper that I've been on. So it's really been amazing because part of my identity, yeah, isn't that cute? Sorry. It's like nerd science, right? Nerd love. So part of sort of like the identity that I have, right, part of the reason that I'm doing the type of science that I am, like I said, is the collection of my experiences. And this is a really important one that to me is really, I cannot separate the science from who I am also as a person, who I've evolved into. Now, at this point, we published this work, and this is where I got my position, like Sarah said, as a fellow, and now I stayed on. And I broadly in the Villeda lab, it's growing over time. We started by studying the brain. Now all of a sudden, I also study like the liver. We're starting to study the kidney. There's some work in my lab now in collaboration with the Sports Medicine Center looking at osteoarthritis in the knee. But predominantly, I look at the brain and cognition. But really, I'm just fascinated by this idea of communication and aging. So now I gave you a lot about like who I am, right? And I hope I told you, and I hope I conveyed to you that starting from where I started, this poor son of immigrants, all the way to faculty at UCSF, it just seems like a crazy trajectory, and it really was a collection of all of these different experiences. Now, the reason that I say this is because in my own research, I'm constantly thinking about, number one, who am I trying to sort of serve long term? And for me, I want to go as broad as possible, which is why I focus on aging rather than any one particular disease. I actually am agnostic to disease. I figure if we keep each other young, we're going to stave off a lot of diseases. But I'm thinking about not just, like necessarily the affluent, wealthy. I'm thinking about what can we do that's tractable for everyone. That comes from my own experience. I'm thinking about these memories, these relationships that I've built that ground me and that push my research forward. I want to maintain them as long as I can, which is why I focus on cognition and really maintaining that identity as long as I can. So all of this is what led me to my research program and led me to the way that I run my lab and the questions that I ask. And what's amazing about research is that we ask the questions. No one tells us what to ask. We get to say, this is important to me. This is why it's important to me, and now I'm going to discover it, and I'm going to do it. So like I said, the main reason, the main question, right, is this idea of can we preserve identity by countering aging? But identity is really hard to model, and I'm a basic biologist. I mean, I showed you, I love newts that got, formed other newts attached to them, right? So I'm a basic biologist. This question of identity is very hard. It's not very tractable for me to be able to sort of build hypotheses and try and figure things out at a cellular molecular level. So I had to refine my question when I decided to start a lab. And what I'm asking more broadly is, can the effects of aging on the brain be reversed. So I know in the end, the brain controls cognition, and part of cognition will be identity. But I have to take a step back, and I really have to go down to the fundamentals of it. So can the effects of aging then be reversed? And that's something tractable that I can actually ask. Now, why do I think it's important? I kind of mentioned that all of you are like either here or still on your way up here, so good for you, you're great, your cognition's wonderful. And I'm really interested in sort of this, which is, you know, kind of in your 70s. It's right when you retire that this starts happening. So I'm really interested in sort of this decline that naturally happens. This isn't something that we're gonna not happen. This is gonna happen to everyone in this room. If you live long enough, it will happen. Now, it also turns out that as cognitive decline is decreasing, increased vulnerability and susceptibility, right, to disease increases. We often think about Alzheimer's and dementia, and those are the ones that are like impactful in our brain because we know we lose ourselves. We are no longer who we are. We no longer identify our loved ones. Our loved ones see us, but we don't see them. So that's really, to me, what I'm trying to go after. However, even if you have genetic predispositions, like even with ApoE, you're not going to have this overt pathology of like dementia when you're 20. So something about your young body naturally on its own, biology figured it out, it's able to counteract it. So I'm fascinated by, can we figure out what's going on here, reintroduce it later in life, and basically just prevent this from happening, right? Maintain ourselves as healthy as possible. I'm really interested in interventions and interventions that are not specific to the brain, but the whole body. For example, things like exercise, and I'll get into that. Now, what do I mean by cognitive decline, just for the purposes of the research that we're actually gonna do? So let's pretend that I own a Tesla. I don't, but let's pretend, most faculty do. So I come in the morning here at Parnassus, I'm gonna park it in a parking lot, and then my brain's naturally gonna look at those cues around the room. Maybe I park close to a view of the Golden Gate Bridge if it's not too foggy, or maybe it's, you know, Pride Month. I come back at the end of the day, and my brain just naturally goes, oh yeah, you park near the bridge, I'm gonna go over there. This is spatial learning and memory. When you're young, no problem. When you're old, you have a problem. And you're gonna see people with their car alarms like lost in the parking lot. Full disclosure, because I've given this talk and I have these like 20-year-olds freaking out because they can't find their car. Chronic stress does this too. So just keep that in mind. So if this is you and you're not late in life, this might be happening. Now, this same type of cognitive decline, spatial learning and memory, it's actually conserved across species. So in order to manipulate, remember that I sewed mice together, right? That's how I started looking into biology. I cannot do that in humans. So we have to model this then in model organisms. And we use the mice. And we use the mice because they actually are pretty good models of aging. If you can see a young animal, shiny coat, nice and lean. You look at the old guy, gray hair, plump. If you were plump, it's a good word. If you were to kind of hold it, its skin is less elastic. If you were to look at, you know, naturally this particular type of old mouse forms osteoarthritis on its own, higher levels of inflammation. It really models a lot of aspects of aging. We even know roughly, you know, like a 4-month-old animal is kind of someone in their mid, 30s. An animal around 2 years is around someone in their late 60s or 70s. So we can really model a lot of aging with this. Now, from my perspective, the brain, just the same type of learning and memory, finding my Tesla in a parking lot, we can actually teach these animals to find a hidden platform with respect to cues around the room. So like this young animal can find this platform just by learning it over time. An old animal eventually does the same thing, but they make more mistakes. You can see here that they have impairments. And then we can start asking questions. In naturally aged animals, what causes a young animal to become old? And then can we take an already old animal and make it young, at least functional? Now, just so you can see kind of what it looks like, hopefully this works. Does it work? Yes, perfect. So this is just the types of mazes that we use. On the left is a young animal. Focus on the sort of upper left-hand corner. You can see that the young animal, even though it's going slower because it's really, really thinking through things, you see it already found it. You can see that the old animal is actually faster. That surprises people. Like when they freak out, they start swimming pretty fast. But you can see that it really does not remember where that platform is, but it will find it. So we're going to spend 30 seconds looking at this just so that you can see that it does actually, in the end, figure out where it is. There we go. OK, you found it. But you can see aging. I just showed you cognitive aging using mazes. So I hope you can appreciate that. We really can quantify this. We just use number of errors. How many times did it go into the wrong direction? And we try and manipulate it such that can we bring back or improve function? And this is just like us trying to find our car in a parking lot. Same exact process that's happening there. So this focuses on one part of the brain known as the hippocampus. Okay, so I'm going to go delve deeper. We're going into the biology now. Now, I mentioned that, in my big question, rather than preserving identity, I went down to, can the effects of aging be reversed? And we have aging hallmarks. We actually know kind of what's going on with aging. We have loss of regeneration, and this is true throughout the body. We have increases in inflammation. This is also true throughout the body. The brain is specific to a particular type of neuron, right, or a particular type of cell, which is neurons. Their communication changes. So before you get disease, they're still there. They're just not very good at communicating. It's not very stable. And then one more thing that we're really appreciating more and more recently is vasculature. We have a blood-brain barrier, but vasculature, what's allowed to come in, the communication between the blood and the tissue through the blood-brain barrier, through the vasculature, really important. That also changes. There's all sorts of stiffening changes that occur. Junctions and receptors and what can come in is also changing. So collectively, all of those changes are leading to that cognitive decline. That's what's happening. So my lab studies every one of the hallmarks, and we try and figure out what combination of these do we have to target to improve things in the brain. And again, I mentioned parabiosis. What I'll mention is that young blood is also really good, and it can actually promote improvement and function. But it's not just about sewing mice together. We already know there's a number of different interventions that are really good. For example, caloric restriction is really good. Even in primates, it can extend lifespan. Exercise, we all know this. If someone, even if they have a genetic mutation, if they're able to exercise, it slows down the rate of decline in your brain just by exercise. So we know this is true in humans. But guess what? As we get older, if you have disease, you're frail. I'm sure you'll often see patients as well, right? Like my mom, who I take, she's just slower at things. It's not like she can do the same that I can and go running. So we know there's this intervention that's really good. Unfortunately, the people that would most benefit from them cannot do it, right? So we focus right now, what I'm going to talk to you about for just a few minutes is going to be on exercise. Now, the question that we had is, can the benefits of exercise be transferred through blood? If you can sew two mice together and get improvement, you can actually just take young blood, plasma, inject it into old mice and get improvement. And young blood and the effects of exercise are really, really similar, and exercise affects the whole body. Then why couldn't it be that your body has information in the blood that can tell your tissues, hey, you're exercising? And we kind of wanted to know, could we kind of hijack that? Are there messengers in your blood that tell your body you're exercising? Could we identify them and actually have the effect of exercise without ever actually physically doing it? So I'm going to give you a summary of some of our research. This shows humans. I use this because it's a cute schematic. We did most of it in the mouse, so I'm going to show you the mouse data. But there's basically the concept. And since then, additional labs have also seen the same thing, so now it's been replicated in different parts of the world. What we did is we took old organisms, okay, and we ran them, and we collected their blood. Then we just took that blood plasma, we injected it into organisms that had never run in their lives, totally sedentary. And then when you added that blood to them, you actually got cognitive improvement. So we could actually get the effects of exercise without ever having to physically do the exercise itself. And that's fantastic for frail, elderly individuals. So now let me show you the actual primary data so that you believe me, because I can show you p-values. So again, same thing. We ran the mice, collected just the plasma, injected, and then did behavior. And I'm just going to show you that water maze that I showed you the video of. We just count how many times does it go into the wrong arm before finding it, and we just average those errors. So it's pretty simple. So on the Y graph here, we just have number of errors. The more errors, the more times you went into the wrong one. So this picture here depicts 3 errors. So in black, we just have normal sedentary, lazy old mice that get blood from lazy, sedentary mice. And in blue, we have those that were sedentary themselves but got that runner plasma. And what you can see is by the testing day, you can see this big separation where the animals that got that runner blood actually had less errors in finding the platform than those that had the sedentary blood, really suggesting that something in our blood is able to tell your body, hey, you're running, and elicit those improvements. And I will say since then, we've been collaborating with a number of different labs, and this holds true also in muscle. It also holds true for inflammation, not just cognition. So it's not a brain-centric thing. It really seems that you can improve a number of different tissues with the substances, right, that are in the blood itself. So really quickly, I'll tell you one factor a little bit and how I'm no longer just in the brain, but starting to look at other tissues. We looked at proteomics. So basically, we surveyed the proteins that are circulating in the blood across a lot of different animals that were either sedentary or running, and we just asked, what's changing? And when we looked at what's changing, most of what was changing was related to the liver. I really expected muscle factors. I just thought, I'm running, it's got to be muscle. We predominantly identified liver factors. We focused on one here, which is just called, it's a factor, you just need to know it as GPLD1. Here I'm just showing you that it goes up in blood, so the darker the bands, each one of these is an animal, and that's their blood, it's just higher. We got to go to the humans though, right? We want to make sure what we're studying is applicable to the human condition. So we got elderly individuals over 70 that had Fitbits for a month, and we collected their blood. And we could actually just say, okay, well, what happens then between more sedentary individuals versus those that are active? And we just used sports medicine metrics. And we had like their steps, so it was really nice because we could do that. And the same thing, this liver blood factor was higher in these humans as well. So we got really excited because that means it's not just an animal thing, it's also in humans. We went and we saw where is it going up, and it's pretty specific to the liver. We looked at other tissues, but it's really increasing in the liver and then going out into circulation. So what exactly is it? Well, it turns out that GPLD1 is an enzyme, and it acts as a scissor. And basically, your body produces different factors, and some of them get a little fat tag called a post-translational modification. It's a GPI anchor. And it basically just tethers the protein to the cell surface right here. So most of the time, they work fine on the cell surface, but we have these proteins, GPLD1 is one of them, and all it does is it just cleaves it. It's like a scissor. And all of a sudden, then, that protein is released. And a few things can happen once we release that protein. That protein can go on and activate different signaling pathways, other cells. It could go on and inhibit cells or signaling pathways, or the protein could just be degraded completely. So it can have a lot of different effects. So we got excited because it does a lot of things. So the first thing we did then is we wanted to know, well, what happens then if we just increase this in our blood? Is that enough to get the effects of exercise blood, which is enough to get some of the effects of exercise? So we're really trying to get down to molecules because that's what we think could be a therapeutic. If we can start making something and dosing something, we think that could then be a therapeutic. And the other thing we did is we made a form of that enzyme that can no longer act as a scissor. So we wanted to know, is it really important for us to cut those proteins from the cell surface? So we stuck it into the liver, and it started producing it in the blood, and I'm showing you again some behavior here. And what I'm showing you is just like, look, number of errors. And again, old animals make a lot at the beginning. They got much better, but still about 2 errors if they have just that normal, their normal liver protein. But then when we increase that liver protein, they actually make less errors. So just increasing this one liver protein in your blood can improve that cognitive function. If you can no longer cleave it, though, however, you're in trouble. Because at that point, then, you can no longer cleave it. So I'll give you a little bit about what we're doing to kind of delve deeper. We love like these puzzles, and we love to start going down to different targets. So now we know, okay, it must be really important to find a factor that gets cleaved. There's about 150 potential candidates. Only about 5 of them we know are actually targets of this, and none of them are like related to anything involving cognition. So we just looked at all of them on all these data sets. We looked at gene expression. We looked at tons of things. And most of these like proteins are either on the immune cells or they're on the vasculature. So we got really excited, and we're like, what about the vasculature? That's, you know, in between blood and brain. We went back to our human data, and we looked at correlations. So here we went to MRI. Now, in MRI, there's these things called white matter hyperintensities that are indicative of like vascular damage. And it turns out that the higher the level of the blood of this protein, the less of those intensities you have. So it's more like vascular integrity. So there's a nice association in the humans. So then we had to go in and start going into like the actual human condition. So from there, we then surveyed all the proteins that have those GPI anchors that are on the vasculature, and we found one factor. And of course, UCSF being UCSF, we have collaborators here. So right down the hall, there's an expert on one vascular protein called ALPL. And it just sits on the surface of your vasculature. But as you get older, there's more of it on the cell surface, and it stiffens your vasculature. It stiffens the blood vessels. So we were excited to know, like, is this then a target? So here I'm just showing you we just put these 2 proteins together, ALPL and GPLD1. If it's a target, it'll get cleaved, and we'll get more of this protein kind of in circulation. And that's all we did. When you put GPLD1, you get higher levels in circulation. You prevent the cleavage. You no longer get any circulation, so we found a target, and it's on the vasculature. So what about in vivo, like in an actual animal? Now, you'll see here in red, these little like squiggly lines. That's your vasculature, and I'm just showing you that protein. And you can see that when we increase that liver protein, all of a sudden you have a lot less of this factor on the vasculature. So we're kind of reducing the levels that normally go up when you're older. And then we did another one. This is activity, just to show it to you in two different ways, because the activity is what kind of causes the problems as you get older. And you can see there's less activity with GPLD1. So of course, we had to go into cognition. I love function, right? So then we asked, okay, well, we went from exercise to exercise blood to a blood liver factor to a vascular target. So what happens then if we inhibit the vascular target in these old mice? And we did that by putting an inhibitor in food. We're trying to get more translationally relevant. One was like gene therapy into the liver. Now we're doing like inhibitor in your food. And this is what the behavior looks like. So here is just GPLD1, just like before. We see a really nice improvement. But when we target that vascular protein, we also get an improvement. So now we think we found this really cool communication between the liver and the brain, where you exercise, you increase factors, they're targeting the vasculature, and these are all different targets that we can try and improve cognition. Now, going back to my original kind of science question, can the effects of aging be reversed? We think they can. We think that there is a number of different of these kind of tissue communications. It turns out that young blood looks totally different. That's like immune mediated. It's your immune cells that are changing. We're doing some work on caloric restriction, and let me tell you, you can transfer the blood of calorically restricted animals and get improvements. So that one's kind of weird because I don't know how I'm going to explain that to people, because I'm going to be like, no, I still want you to eat normally. I don't want you to be fat because it doesn't take care of the fat part, but it does improve function. So the cognitive one with caloric restriction, that seems to be more fat and kidney, and then exercise seems to be more liver and a little bit of muscle. So each one of these interventions that we know are really good seem to be mediated by different communication from different organs, and we're really excited about that. Now, my big question, right, can we preserve our identity? So long term, now that we're finding these factors, we're absolutely trying to get them now towards the clinic, right? We're trying to develop small molecules. We're trying to look also at recombinant proteins as well as inhibitors. And we're starting to do some also studies in terms of toxicity. And the idea would be, can we leverage these factors, give them to the elderly individuals, maintain their function for as long as possible, prevent disease, prevent Parkinson's, prevent Alzheimer's, really prevent any age-related disease. And with that, I'll just thank my lab for all of that. So that's sort of where we're at, trajectory. From who I am to what I'm doing here at UCSF, I think I went over by a little bit.
Thank you so much. So we have a little bit of time for some questions. Thank you so much, Dr. Villeda, for this wonderful talk. I hope people enjoyed it as much as I did. I'm Stuart Gansky, our Associate Dean for Research. So do we have any questions from the audience?
Hello? Okay. Firstly, thank you so much for such an interesting presentation. I did write down the question because I'm old and I think my cognition is also going down. Is it so that the kind of research you're doing is something where I think there is this famous personality by the name of, what's his name? Yeah, Byron Johnson, who is basically using, I think, his son's blood as transfusions to keep himself looking young and feeling young and reverse the aging process. So I guess my question is, firstly, how long does one have to keep doing this to start seeing results? And also, how practical is this on a large scale, assuming all of us want to reverse the aging process?
Very good question. Full disclosure, FDA already said this, blood transfusions is not the way to go. It is not tenable. It can actually be deleterious, particularly like repeated transfusions with the pressure around your alveoli in your lung can actually cause serious problems. Heaven forbid there's any sort of infection with the blood. So not tenable. Even if we wanted to, there's not enough blood today to treat everyone with just Alzheimer's disease, let alone this like silver tsunami that's coming. So no. So the FDA already said it. If anyone tries to sell something, tell me, because I will email them and I'll try and get someone to like be kind of, no, that's just, we write opinion pieces about this all the time. This is the reason why we're trying to find molecules, mechanisms, right, molecules, so that we can actually then do it in a way that we know, with clinical trials, with dosing, things that are regimented, placebo, double-blinded. We're getting there, and there's a number of like different factors that have been identified at different stages. But absolutely, that's not anything that I would recommend. Obviously, there's these N-of-1 studies that you can't stop, but no, under no circumstance would I say that's the way to go. Yeah.
Thank you.
And there's one here too.
Mike, or if-
Yeah.
Can I-
the person
Someone
who's
else
standing
first?
up in the back there. I think that was the first hand I saw.
Hello, hello. I have a question. So you use the water maze as your main form of measuring like the cognitive.
Yeah.
Is there other ways of doing it? Is that just the industry standard? Like everyone who does research on this is using this water maze, or is this what like your own lab's technique?
No, we use, at least 4 different cognitive tests. It doesn't, this one's really good exercise and it impacts all sorts of memory. This is spatial memory. It does working associative. For the purposes of like talks, it's just visually so much easier to see it that I tend to use this in my talks. But in the papers, there'll be usually 3 to 4 different behavioral assays. And it gives us specificity.
Thank you.
Another question here.
Yeah, I'm just going to ask. Perfect. Thank you so much for the talk and also sharing your story. It's really inspiring. My questions are 2. The first one, I'm curious about the dose that you gave for these experiments. I'm curious, is there a dose response that improves the cognitive function?
Yeah.
And then 2, is there a tolerance that's built up to this? Like if you're dosing the animals for a long time, does their cognition stay the same or does it actually get worse or improve?
Yeah, really great questions. So we do about 5% of volume for plasma, and we do that every 3 days for about a month. So they're short-term treatments for this. If you stop the treatment, you still see the effects there 3 months later. By 6 months, the effects are gone. So it's not that you need constant exposure to something in order to maintain that going on. For the dosing, what we've tried to do, because we did a more gene therapy delivery method with the exercise factor, is we merely just raised it up to the level that we saw with exercise. But right now, we are making the recombinant proteins and doing that sort of dosage. We have other factors, like young blood factors, that we've already gotten to the recombinant protein phase. All of them, though, it will go away if you stop dosing. Yeah. And I'll be in the back.
Yeah, so I think in the interest of time, I know there's a lot of excitement and questions. I actually have some as well. But we're a little behind schedule. We're scheduled to have a break and return at 10:15. So I would suggest that we try to be back here by 10:15 so we can get back on schedule. And then please, you can ask Dr. Villeda your question. He's going to be around a little bit longer. So please join me in thanking him for a wonderful talk again.
Should I get people to get back to their seats now?
Yeah.
All right, everyone, if you could start making your way back to your seats, please. How you doing, Stuart?
Yeah, and then just so that it goes on to the screen, you mentioned set that just present, and then because this is set as the background, essentially. If that makes sense.
So what do I do? I exit,
So you
escape,
have
and
to,
then
you have to escape, and then you'll click on the next one. I can come up and do it.
I can do it too. And just,
Yeah,
so
you
just
said
escape
escape,
and open up the
and
next
it'll
one.
leave it up there. Yeah.
I
and then-
see.
Yeah. And you just click on that.
Oh, never mind then. All right, everyone. We do have a lot of seats up front. So if you were standing in the back, please make your way forward to the front. All right. Welcome, everyone, to the morning session for our oral abstract presentations. My name is Ricky Lugo. I am an assistant clinical professor here in the Department of Oral and Maxillofacial Surgery, and I'm very excited to see all of the wonderful work that our students have done. Thank you. We do have a few seats up front, so for those of you that are standing in the back, please feel free to make your way forward. Our first presenter for the morning is going to be Roaa Saadeh, and presenting her presentation is entitled The COVID-19 Pandemic Impact on Medi-Cal Adult Dental Utilization under the guidance of her mentor, Dr. Elizabeth Mertz.
Hello, everybody. It's nice to be here. As was mentioned, I am a first-year dental student, and I had the privilege of working with Dr. Elizabeth Mertz and her team at the UCSF Health Force Center at UCSF, and I will be examining the COVID-19 pandemic impact on Medi-Cal adult dental utilization. Now, before I begin, I would like to provide a background for my research, which first includes defining what Medicaid is. So Medicaid is a public health insurance program, and it is the most used public health program nationwide and is used by millions of individuals. Each state has their own rules when it comes to who qualifies for certain care and privileges, and California is one of those states that have extensive benefits for their adult populations. And the Medicaid program in California is particularly known as Medi-Cal. So it goes without saying that the COVID-19 pandemic has exacerbated a wide variety of health concerns and has caused disparities, and the dental health workforce is no exception. We saw a large number of closures of dental practice, as well as the pushback of a lot of non-emergent procedures. So with that, because the dental public health programs had to closely follow so many guidelines, I hypothesized that this impacted Medi-Cal adult dental utilization. Now, I have three specific aims in my project. First of all, I was aiming to quantify adults' access to dental care in the Medi-Cal dental program before and after the COVID-19 pandemic, describing the enrollment of dental providers, and then describing other COVID-19 pandemic impacts on the adult utilization. My methods include mixed variety. First of all, I retrieved my quantitative data from the California Medi-Cal Dental Claims, the American Dental Association Health Policy Institute, as well as other nationally publicly available sources in comparison. In addition, my qualitative data was retrieved from the interviews I conducted with dental public health leaders and a literature review. And the main findings of my research project came to be that there was a decreased utilization of Medi-Cal adult population usage, and there is a disparity, particularly in different settings. So when we look at fee-for-service settings, which are typically the usual private practice that bill providers based off the number of services that they give, although we did see a decrease in the utilization, it does not compare to federally qualified health centers, which are intended to increase access for care for patients. So we will be seeing more of the details in the following models. But first of all, I specifically looked to highlight 5 counties within California so we can observe these trends. The 5 counties we will pay particular attention to are Del Norte, Los Angeles, Orange, Plumas, and San Diego. Now, this is one of the interrupted time series regression models that my team and I developed. Not to fear, it's complicated, but it's very easy to follow along with. So first of all, if we observe the first trend, that describes the pre-COVID trend of adult dental utilization. So in fact, there was an increase in adult utilization, although we can always make improvements in our field, there was a positive trend. As soon as we see those red dashed lines, we see the impacts of the COVID-19 pandemic mandates and how that impacted dental closures. And of course, we saw a very significant reduction. Following the uplifting of mandates, we saw that there was, again, a positive trend. However, the previous usage and utilization of dental care never really returned to how it previously was. Now, this is for fee-for-service settings. What if we looked at federally qualified health centers? So we see the same trend. Prior to the mandates of the COVID-19 pandemic, there was, yes, a positive increase in utilization. There was a severe drop in utilization, but in comparison to the previous model, we see that there is still a drastic reduction that never really recovered similarly to the previous model. With that in mind, the second aim that my team and I also were focusing on is examining how dental providers utilized Medi-Cal during the COVID-19 pandemic. And it was not unsurprising to us that there was a decrease in the number of new providers that were accessing Medi-Cal and utilizing it for their patients, as can be seen in the chart right here. And in addition to the models that we have developed, as I had mentioned, I also conducted interviews with dental public health leaders in order to examine why do we have these results and how can we contextualize them. Well, we were able to summarize 3 main points as to why we were able to see these trends. First of all, it includes the disproportionate workforce issues on health centers. So there is a disparity in terms of usage and resources available for fee-for-service programs as well as federally qualified health centers. Especially with the COVID-19 pandemic, the closure and resources that were available, there was some disparity. Second of all, the hesitancy and concern of dental patients. So the COVID-19 pandemic drastically changed the way that we perform dentistry. One specific example is the extra usage of telehealth in dentistry, which changed the access of not only patients, but the way things were done. And then finally, institutional and organizational constraints. So there is still a lot of work to be done in order to bridge that gap in access to care for patients. And this is one of the reasons why we see such disparaging differences. Now, I have these following figures in order to further highlight the differences between fee-for-service and federally qualified health center settings. And as you can see in the figure above, you almost see a jump back in the utilization of care. However, for federally qualified health centers, we can see that disparaging effect is still in effect. In conclusion, although the counties in California, and the particularly highlighted ones, the 5 counties I had talked about before, did not return to their previous pre-COVID-19 mandate levels of utilization, there's still a lot of work to be done. And this research is in support of making sure that there is focus in investing in health policy research, as well as ensuring that we are addressing oral health disparities in California. The research that we hope to continue is further examining dentist enrollment in Medi-Cal and perhaps, being able to compare between public versus private sectors and so on and so forth. With that, I would like to thank again my faculty mentor, Dr. Elizabeth Mertz, her incredible team at the Health Force Center at UCSF, as well as the Philip R. Lee Institute for Health Policy Studies. And I would love to answer any questions anybody may have. Yes, please.
So thanks for a really great presentation, very clear and excellent work. I was wondering if you could give us a little bit about the rationale for selection of those 5 specific counties that you looked at.
Yeah, absolutely. So the intention of course, all 58 counties within California were represented in our research, but if we look towards the maps, we can see that the counties come from very, not only different geographic locations, but there is major differences in terms of the income level, the racial population, and so many other factors which are included in our covariates when we built our models. So you can see a county such as Del Norte, it doesn't necessarily have the same financial comparison as Orange County or Los Angeles, in which the income level is greatly different, the racial population is different, which also impacts the access to care generally, as has been seen in different public health trends and whatnot. Thank you. Yes.
I noticed in your, if you go back to the first graph that you showed, there's a slight increase in utilization just prior to COVID. I'm wondering if you can speculate as to why that is.
Yeah, so this is something that we looked at previously, and this graph particularly covers the time period from 2019 up until around 2021. We have some conjecture as to why that may be. One thing could be because the amount of people that were seeking care, it fell around towards the end of the year, therefore they want to utilize their benefits then, or perhaps it's because people just wanted to get their care done, knowing that it would, the pandemic would impact things, and it was just something that was expected. But definitely more research needs to be done to pinpoint exactly why we see that increase in utilization prior to that drop. We can compare it to federally qualified health centers too. We do see that small increase, but more research should be done in that area. Okay, thank you very much.
All right, next we have Andrew Lee presenting on ex vivo application of an innovative 3D printed access guide with CBCT and pulp canal obliteration endodontics treatment.
Sorry. Hello, everyone. It's my honor to be here speaking to you all today. I'm here to present my projects. I actually present this project at the National Endodontic Conference this year, and it's my honor to be here and share this project with you all. My name is Andrew. I'm currently a fourth-year dental student. I've been working with Department of Endodontics for the past 2 years on this guide design. My research project is trying to focus on treatments of calcified canals. And I'm trying to create a guide that is not only new when it comes to design, but also easily accessible for endodontists to make. Okay, I know I have a diverse audience here. We have D1s, D2s, and also clinicians. That's why I would like to spend some time going over what a root canal treatment is. Usually, a root canal treatment involves mechanical debridements and chemical debridements of canal systems. And the most important thing when it comes to going to the root canal treatment is to get the proper access so you can go into the canal systems. And to be able to do that, for those clinicians that place implant, you probably heard something called implant guide, or probably used an implant guide. And that's why really when it comes to early 2000s or the mid 2000s, that's when the implant guide, people start to thinking about using that for epicoectomy, for microsurgeries. And from 2016, more and more people start to thinking maybe we can use that for accessing the canal location so we can accurately locate the canal orifice. And when it comes to guided endodontics, usually there are two workflows. One of them is a little bit newer, which is the one that I'm mainly looking at. Well, the traditional workflow involves a use of intraoral scan along with a CT scan. And for simplified workflow is exactly what I mean. We're trying to bypass the intraoral scan. And this is a little bit of a newer concept. And this is something that we really trying to work on and look at, because for endodontists, a lot of the endodontists do not have intraoral scan in their office. And this is really the biggest roadblock for guided endodontics. So that's why we're trying to utilize the intraoral scan, utilize the CBCT and simplified workflow only. All right. So before I go into my project and my guide design, I have a little bit of a literature review on guided endodontics. So this study is actually published in 2020. It was done by the University of Connecticut endodontic residents. They're using CBCT scan with the SIGCAD software to design their guide. And then you'll probably see this is the picture of the guide, what it looks like. They actually also use a intraoral scan. And then the guided canal really just 3 access hole there. They're trying to pinpoint and tell the clinician where the canal orifice is. But you can probably notice that there's not a lot of guidance going on. There's no sleeve. When you're going into the canal, most likely you have to take out the guide and still make your traditional regular access. And for calcified canal cases, they didn't tell you how deep you wanted to go. And they do have 40 sample size, and do find out that the access time is a lot shorter, which also a lot of us already know having a guide make the procedure time a lot shorter, and that's why this is a promising area we want to look into. And this is a clinical case that's being done in 2022. And if you take a look at the picture, this is if the clinician here places implant, this is more like a traditional implant guide that people are using. But they combine this with a sleeve, so they can properly guide the drills into the canal systems. And this is the clinical trial studies. And They successfully located the canals, but they have to, while they're doing the access, they have to take out the rubber dam. They have to properly find the canal location first before they can proceed with their regular endodontic treatments. And I included this in there because this is a traditional pathway of doing guided endodontics, and I'd like to share with you all like how our guide design looks different compared to this. And before I go into my official guide design and my research, this is another study published in 2021, and this is something I think is very, very interesting to share because it's not like a traditional surgery guide for implant. They actually use an inlay guide because the tooth is so bummed out. And in this case, is really, really interesting because they have a fused distal canal. The location is very abnormal. And they tried 3 clinical visits. They couldn't find the canal location, so they ended up having to create something like this. And that's what I think where endodontic, for especially guided endodontic and static guide, where I think this is very important to clinicians and can potentially be really helpful for cases like this that are very, very difficult to find, multiple visits fail, and that's maybe the time for you to think about maybe I can create a guide to try to find the canal locations. All right, so now I want to go into how I perceive my research and how I want to design my guide. So I want to have a newly designed guide that bypass intraoral scan. I want to utilize what we call a simplified workflow, and I want to potentially make this systematic. So I introduce a 3D printing teeth stimulation prior to we actually doing on it is the actual extracted teeth. So we have a 3D printing teeth, and we have an actual clinical ex vivo extracted teeth that we want to do. And we also video recording them to try to see how well the clinician used the guide and how much longer it takes for them to access the canal systems. And yeah, and hopefully we can potentially make this systematic and we can extend this outside of just academia usage. All right, so this is just some example. We have multiple teeth that we're trying on, but this is just an example of the teeth that we picked to conduct our trials. And you probably see a lot of those teeth are calcified, or those are really, really difficult to treat when it comes to endodontic treatments. A majority of those teeth, you have to trough them to be able to find a canal. And that's why we're trying to focus on it. Those are the type of teeth that we pick when it comes to these studies. Oh, and then we're trying to go into our guide design. So our guide, when I go into it, I know I want something that is easily accessible. That's mean I want to think about how endodontists approach the appointment. So they usually already have rubber dam in place. They want to just go in there, go into the canal. I don't want them to access it, put on the rubber dam. It would just take too long for them to do. It's not quick enough. So I have a guide that I designed. I have interproximal clearance. I have 1.3 millimeter access when it comes to the canal sleeves. And then I have a 3D printing teeth that we print before we're going into the actual teeth. And we have 3 different designs. We have straight line access design, which is from A1 to A3. You can see the X-ray there. And we have a converging access design, which is B1 to B3. You can probably see the drill path is actually crossing. And we have a C4 to C6. You can see that it's a single canal guide design, which we using metal sleeve support, which because we have that metal sleeve, we use a lot of resin, so we can only find 1 canal every time when we use this one guide. So if you have 3 canal, you need 3 guide. All right, so here's our result. So for straight line access design for A1 to A3, you can probably see because we're trying to test out how accurate the simplified workflow is. We don't want to use intraoral scan. That means we definitely want to make sure that we have something that is accurate, so we use straight line access. But you can see, because we're trying to focus on getting the straight line access, we end up compromising a lot of the important tooth structure that dentists wanted to preserve when it comes to accessing. So we know that straight line access design is not going to be the way to go. And we have our convergent access design. It was clinically acceptable. We located all the canals, but the only problem is that toward the end, because the drill paths are converging, and then we don't have a metal sleeve, so we end up compromising a lot of the resin that we're printing out. So toward the end, we really don't have a lot of guidance going on. And our very last guide design is a metal sleeve support. You can see because we have a metal sleeve, so we're using a lot more resin, but it was a lot more stable. We're able to find all the canals with very stable and accuracy guided endodontics. All right, so this is our recommended method and workflow. So we recommend it for the future. If we want to do a large clinical trial, we recommend a patient consultation appointment first. We get a CBCT scan, and then we send the CBCT scan to the 3D printing company. So we print out the 3D printing teaser. You can see the picture there. And we test out the accuracy of the guide before we proceed with any actual clinical treatments or extracted teeth treatment. So that would be A1 there. And I think it's very important to do case evaluation because sometimes if you have a full metal restoration, it's really hard to bypass those noises. So this is something a clinician want to consider if we want to make this guide in clinical, using this guide clinically. All right, so this is kind of like summarized. This is like a summarized workflow of how we finish our study. So we end up choosing the single canal with metal support because it gives us the most clinically accessible outcome and accuracy. And we have a burr that we find out currently all the burr that in the market, we're using Neoden 1.3 millimeter fixation drill, which is not ideal for enamel drilling. So we really run into a lot of problems for that. So we recommend a design of our own burr block to be able to pair this with the systems. All right. So this is just something I want to include because this year, we started working on the studies last year, and just this year, January, there is an article published in Journal of Endodontics there. It says that simplified workflow is just as accurate as traditional workflow, which is really make us have confidence in our research, and then we feel comfortable proceeding with more studies and more trials. Yeah. So what does it mean for our study? Like I said, this shows that we are simplified workflow is just as accurate, just as useful. And then our difference is that we use pulp canal system. We use the calcified pulp canal system. We located all the canals, and it really makes us a front runner when it comes to simplified workflow of guided endodontics. And we also have variety of extracted tooth samples. We have premolar, we have molars. So I'm hoping that in the coming years I can make this more systematics. Yeah, in terms of future directions, how do I want to proceed the studies? I'm a fourth year, so I'll be leaving UCSF, but I will be continuing my education at Harvard. So I'm hoping that I can, during my residency, I can finish my systematic design. I want to continue working with endo department here. I want to make this systematics. I want to conduct more case report, clinical trial. And I already have a burr block that we designed. We call it the ADS burr block. It's conduct of my initial, Dr. Sabetti's initial, and the resident I've been working with. So we're hoping to have this burr block made, and hopefully next year we'll have more result and study to share with you all.
One question?
Yeah, if there's any questions, I'll be taking them. Yes. Yeah. Yeah. Yeah, so usually when you take your CBCT scan, you want a conversion into STL file, and that is the most important thing when it comes to designing the guide. And then if you have a metal restoration, you have a PFN crown, you have a lot of amalgams. We do have a few extracted teeth that have amalgam on there, but we noticed that when you have too much amalgams, you probably need a you probably need an intraoral scan because the noise is too much. But one thing we're thinking about doing is that because when you take a CBCT scan, you also capture the adjacent teeth. So we're hoping we can get guided support from the adjacent teeth instead. So you kind of broaden our scope of patient pool that able to use the systems.
One more question.
Okay, sorry. So I guess my question is, would the stent be able to tap into the presence of accessory canals, and would there be some amount of accuracy in that, or are we just going to be?
Yeah, so for this, it's mainly, so when we go into this studies, we know we want to pick teeth that are calcified. And one of the criteria we're looking at is 4 millimeter below the CEJ. So for those treatments, when it comes to accessory canal, it was really probably all the way down the apically, and it's really hard to get the file go in there. So our main goal is to create accessible access, so we can provide an accurate measurement to the clinician. So you want to introduce your burr all the way down to what length to be able to bypass that calcified area, and then you can get your proper patency from there. And then if you have proper patency, you have proper angulation. I don't know how easily you can treat accessory canals, but I think it will be still be very difficult. But that's when something called biolace or gentle wave can potentially help treating those cases. But I think getting the proper access angulation is definitely the first step of getting clinically accessible root canal treatments.
Thank you.
Thank you. Thank you, Andrew. Next, we have Carson Gadler. Carson Gadler is presenting on actions of combined PTH and calcimimetic treatments and the calcium-sensitive receptor on skeletal anabolism and/or fracture healing under the guidance of Wen-Han Cheng.
Right. Good morning, everyone. So I'm Carson. I'm a D1. And I won't repeat the title. I understand it's a bit of a mouthful. But the basic way you could think about my project and what we were doing is we explored better ways to improve bone fracture healing. And the reason we did this is because osteoporosis is a devastating condition that's consistently rising in elderly individuals. And when these patients experience bone fractures, they result in drastic complications. And I have some of those statistics here. So a year after fracture, 40% are permanently disabled, 18% die directly from the fracture, and even among those who survive, 80% are unable to perform a key activity of daily living. And unfortunately, the current treatments for osteoporosis aren't that great, including the FDA-approved regimen of administering parathyroid hormone, or PTH, to patients. So PTH only has modest anabolic effects and can even induce hypercalcemia in some patients. However, what the Chang lab has recently discovered is that when you take a calcium-sensing receptor agonist called R568 and you combine it with PTH, it helps to synergize the anabolic effects of PTH while reducing its hypercalcemic effects. So knowing this, we sought two main aims. Aim one was to determine the action of the calcium-sensing receptor in PTH-based therapies, and aim two is to demonstrate the utility of a long-acting PTH analog to further amplify anabolic effects in the fracture healing of aging mice models. So the way we did this is in aim one, we took mice with knockouts in the calcium sensing receptor in osteoblasts, along with their control litter mates, and subjected them to a 4-week treatment of either vehicle, PTH, or PTH and R568. And after this treatment, their distal femur and tibiofibular junction were analyzed using micro-CT. For aim 2, what we did is we took 23-month-old aging mice, induced a right tibial fracture in them via 3-point bending machine, and then subjected them to a 4-week treatment of these following treatment groups, after which their fracture calluses were analyzed via micro-CT. Additionally, we took retroorbital serum samples from these mice to measure the calcium levels in their blood in response to acute drug treatment. Okay, so after conducting these experiments, these are the results that we got. So we could see that in the controls, PTH and R568 increase the overall amount of trabecular bone as well as the thickness of the cortical bone. However, when you knock out the calcium sensing receptor, these effects are completely abrogated. And it may be a little difficult to see in the images, but we have quantitative measurements to back that up as well. So we can see here in bone volume over total volume in the controls when you add PTH and R568, they increase relative to vehicle, but when you knock out the calcium sensing receptor, these effects completely disappear. Okay, and so for aim 2, what we have here is these are images of the fracture calluses. These bottom images are the mid-sagittal slice. So what we could see is that PTH, as expected, produces moderate anabolic effects compared to vehicle. However, there's a tremendous increase in the LA-PTH treatment groups. And when we look at bone volume over total volume, we can see they're 100% greater than vehicle on average. They also produce around the same amount of bone. The key difference, though, is that when you add R568, we can see here that it helps complete the bridging of the fracture callus. And also in our serum samples, we can see that when we add R568, it normalizes levels of hypercalcemia, which are seen in LAPTH alone. All right, so what we can conclude from all this is that, one, osteoblastic calcium sensing receptor is critical in mediating the anabolic effects of PTH-based treatments. Two, the prolonged activation of PTH receptors by long-acting PTH can further increase anabolic effects in aging calluses by 100% compared to vehicle. And that combined PTH and calcimimetic treatments like LAPTH and R568 represent a promising new treatment for osteoporosis patients. And this image also sums up the results in AIM-2 pretty well in the study in a nutshell. Okay, so our next step, we want to build on AIM-2. So what we want to do is we want to take the non-fractured bones from these mice, including the mandible, and analyze them under micro-CT to see if there's any bone loss or bone reabsorption from these areas caused by the treatment, which could be used to repair the calluses themselves. So this is a problem in other osteoporosis treatments. We want to make sure that this isn't happening in our treatment. Okay, so I would like to thank the UCSF School of Dentistry for providing funding for the Summer Research Fellowship. Additionally, I would like to thank my mentor, Dr. Wenhan Chang, and a senior research staff member, Dr. Zicheng Chang, who worked really closely with me throughout this project. And these are my references, and that's it. If you have any questions, you're more than happy to answer. Yeah.
The blood that you sampled was, you said retrovulvar?
Retroorbital,
Retroorbital.
yeah.
And so I'm wondering, is there something specific about the blood that comes from the retroorbital region that is different than something from the periphery, like an extremity?
I just know the reason that we did it is because it's the most humane and the easiest way to do it. If you put the mice under anesthesia and you do it, if you do it properly, it won't hurt them, and then they could live longer throughout the whole experiment. In other areas, you could take it from the tail too, but that's really difficult and requires a lot of skill. So that's why we did retroorbital. Yes.
I was wondering on the control when you look at the quality and both quantity, you have considered control with what's the age, what I don't remember the last name. But there was a small hole in the bone. Is it representative of like every single mice you had, or was it just
Yeah, so we didn't. Summarize the question. Okay, so actually, I'll go back to the.
There wasn't a microphone.
I'll go back to the slide. So where's? Yeah. So right here. So this hole, right here. Yeah. So we didn't see this in all the bones that were analyzed. We're not quite sure why this is here, but even if you take this and you analyze it, the structural parameters of it, the overall thickness and the overall amount of the cortical bone is still increasing. We're not sure why that's there, but we could run more trials to further explore that.
Have you seen it in every mice that was treated with the PTH 134, or was it just you chose this one and then had it?
No, I just chose this one. Not every single one is like this, yeah. All right. Thank you.
All right, thank you, Carson. Next, we have Anubhuti Misra. Oh, you're right here. Anu is presenting on To Dare or Spare, the caveat of extraction in post-radiation cancer patients, under her mentors, Dr. Christina Sanchez and Dr. Jennifer Perkins. I'll let you know when you have a minute left. I'll let you know when you have a minute left.
Okay. Hello. Hi, everyone. A very good morning to the UCSF family and a very warm welcome to all the guests who are joining us today. My name is Anubhuti Misra. I'm a resident in the GPR program. And thank you so much for being here and giving us the time and opportunity to present this case. During the last couple of months when I was rotating through dental oncology clinic in Mission Bay, I was reminded of how important it is for oral health clinicians to learn how to manage and treat patients with head and neck cancer. So today, I'm here in front of all of you to present a case of extraction in a post-radiation cancer patient. I would like to thank my mentors, Dr. Christina Sanchez, who was a GPR program director, and Dr. Jennifer Perkins for helping me prepare this clinical case presentation. Okay, so as future dentists, future oral healthcare professionals, what is the one most challenging thing that you could think of? Any guesses from the audiences? Sorry? Yeah, so I would say diagnosing, treating, managing, and cross-collaborating with the other healthcare professionals for patients who have head and neck cancer. So here we have this, cartoon showing the challenge, the heavy weightlifting that we had to do. The background for the case is, out of 2 million people who will be diagnosed with cancer in 2023, roughly 4% of the people will have head and neck cancer. Radiation therapy is the mainstay of head and neck cancer patients, with 75% of them receiving radiation therapy. And it also puts them at high risk of getting osteoradionecrosis. Therefore, it is important to make informed choices and evaluate your risk factors correctly. So this is a slide showing a patient undergoing IMRT, and the other slide is showing a CT showing the tumor. So like I talked about, it's all about interprofessional and cross-collaborative approach. We start with the radiologist, you collaborate with the radiation oncologist, the medical oncologist, the prosthodontist, and the list goes on. So we had a patient who presented, this 42-year-old Hispanic female presented with a maxillary spindle cell sarcoma involving the right maxillary sinus. Her chief complaint was 4 months of fluctuating but overall worsening right cheek swelling and pain on the upper right area of the sinus. Patient went to see dentist, went to the ED departments, to a couple of hospitals. She was given antibiotics with no improvements. Until she was seen in the ED and somebody decided to take a CT scan. The CT demonstrated an aggressive mass in the right maxillary sinus, as you can see, measuring roughly about 3 centimeters. And then the patient went maxillectomy, lateral rhinotomy, followed by radiation therapy, which was completed roughly about 23 days before she was evaluated by dental oncology for pain on tooth number 5. Let's talk a little bit about the radiation therapy this patient received. So the therapy was completed on 8th of August of this year, started on 22nd of June. The tooth we extracted, the root tip number 5, was done on 31st of August. The radiation dosage was in total 6,600 centigrade, which is actually a very less radiation dose for head and neck cancer patients. So she got pretty lucky here. So she was given this dose, total dose in about 200 centigrades and 33 doses or increments. So this is a picture of the case. Coming back to the dental part of it, the dental chief complaint for this patient was that she was having pain in that number 5 root tip, was not getting better with the medications she was getting. She was already in so much of pain, so she came to see us and we wanted to help her. So this patient reported pain 5 on 10. As you can see from the intraoral picture, there was a root tip. Clearly it's unrestorable. And we saw some sporadic focal point ulcerations. There was some mucositis in the patient's mouth. She was also wearing an obturator because she has had maxillectomy and rhinotomy in the past. So this is what we saw. Our assessment was that clearly this is a tooth that needs to be taken out, a retained root tip, and prognosis was favorable with completion of the recommendations. So here, this is the root tip that we took out. This unrestorable tip was extracted atraumatically, and we are going to talk about why it was important for these patients to have as atraumatic extractions as possible later in the slides. And the post-operative course of 2 weeks was unremarkable. Patient had a healthy granulation tissue, no bone exposure, no pain and swelling, and she healed pretty well, and we'll talk about that in the coming slides too. So this is a picture post-op 2 weeks. Apologize for the quality of picture. It's, I don't know if you can see this area. You can see a shadow of the mirror image of the granulation tissue. There is some swelling over here, but this is just post-op 2 weeks, and patient reported that she was feeling better after 2 weeks. Post-op 5 weeks, we checked the patient. There was no pain and swelling. Patient was healing well. She actually, felt that she has healed pretty well and didn't feel the need to come and see us because she wanted to focus more on her sarcoma treatment. She was still going and see the other doctors. So, yeah, but we were at least happy that we were able to help the patient, in managing the tooth pain so she could focus on her sarcoma treatment. All right, so we are going to talk about learnings from this case. The first one is this extraction was under the golden window. So now the question is, what is a golden window of extraction in head and neck cancer patients? So first 4 months, usually 4 to 6 weeks after radiation is the golden window. That's where the radiation effect on the cells have been to the minimum extent. And you can, if the patient needs extraction, as soon as possible, even in the golden window. So that's the recommendation. Sometimes patients are recommended to extract the tooth with HBO, which is hyperbaric oxygen therapy, which also helps in healing in some cases and not all. So this is a timeline. This is the golden window, latent period, and then there are higher chances of onset of OR end moving forward. Second is, we got a successful outcome in this case because the radiation dose was way less. So usually we have 50 osteoradionecrosis, which is the most potent side effect of extractions in head and neck cancer patients who have received radiation therapy, is at the dosage of 50 to 70 gray. And that also, the likelihood is more in mandible as compared to maxilla. So in this patient's case, the tooth was in maxilla, it was a root tip, it was taken out atraumatically, and then the radiation dose was 6,600 centigrade, which is way less than the danger mark here. Also, it is very important for us to know how much radiation is being given to the patient, where the radiation is given to the patient. The patient could have chief complaint in one of the tooth on the upper left, and the radiation is given on the floor of the mouth area. So definitely you can evaluate how much was the radiation dose and if the patient is a good candidate to go ahead and do the extraction or any dentoalveolar surgery if needed. Moving on. So with this case, we also demonstrated how important it is to have effective multidisciplinary team communication and interprofessional patient care coordination. So we collaborated with head and neck surgeons, radiation oncologists, prosthodontists. Also, the patient is not undergoing chemotherapy, which is something we should definitely consider. If patient is having concurrent chemotherapy with the radiation therapy, patient is likely to have damaging effects worth 1,000 centigrade. So that is an additional dose you should take into account before you decide what is your treatment plan. And fourth is atraumatic extractions. So remember, we talked about why it's important for these patients to have atraumatic extractions. Atraumatic extractions basically means your body has to work less for it to heal. There is, if there is primary closure, great. If not, then body has to work extra hard. There is greater chances of food lodgement. Bone healing is difficult. So basically in these patients, atraumatic extractions is the key. And if you're not able to, then that comes with the risk. But obviously then if your risk and your benefits outweigh the risk, then you take a informed decision over there. All right, questions about this case? Yes, please.
What made this? Is that work?
Use this one.
There it is.
Is it? Yes, please. So you said that this patient was lucky enough to receive low dose of radiation. What made this patient receive this, like you said, 6,600?
Centigrade, yes.
And what is the average radiation in a head neck cancer normally?
Yeah, thank you very much for that question. That's a great question. So first of all, we don't decide the radiation dose the patient is supposed to get. That depends on a lot of factors. How big is the tumor? Where is the tumor? Is the tumor more amenable to chemotherapy, or is the tumor more amenable to radiation therapy? That decision is taken by the tumor board. There's a board which consists of all these doctors and specialists, and they make that call. So the patient got lucky because in her case, they decided to limit the radiation to this dosage. The second question, what was the second question, sorry? The average, okay. So that also really varies from case to case. If you have a tumor in the body of the mandible, that's definitely, and if it needs radiation therapy, it's definitely going to be on the higher side. So really varies on case to case. What's your radiation therapy? Some patients need chemotherapy, some patients don't. Some patients need chemo and radiation both. Some need surgery first, even before they can get to radiation and chemo. So that's really decided by the tumor board, and that's why it's very important to collaborate with different health professionals. There's also this code, I forgot to talk about it in the presentation, which is billable by Medi-Cal. Sorry, just taking a couple more minutes here. Yes, D9992, patient care coordination code. So that is something we use in dental oncology to bill the patient and the insurance for this. So yeah, I hope that answers your question. So any other questions from the audiences? Yes, please.
So I just want to know how the treatment plan would have been altered if the patient was not within the golden window. Would we still proceed with the extraction? Would that then predispose the patient for osteoradionecrosis?
Yeah, that's an interesting question. So we always talk about risks, benefits, and alternatives in our cases. So if the benefit outweighs the risks, then we will go for it. Also depends on how much of how much of pain the patient is in, or swelling, or if there's an infection. So how much it is really bothering the patient. The overall aim is to improve the quality of life of the head and neck cancer patients. And if that is something we can work with, given X amount of radiation dose, if it's going to significantly impact improvement in the patient's quality of life, we will go ahead and do it. We always tell them, we give a full disclosure, you are at a high risk of ORN. Some cases, we won't do it. The other alternative to extraction is coronectomies. I don't know if some of you know about it. We basically request the patient to get the root canals done, and we remove the crown of the tooth from the gum line. So patient is able to maintain that kind of bone over there. Bone is not injured or touched. We don't touch it with the risk of ORN. We just let it stay there, and we cover that root tip with a composite so that it doesn't get any cavities. So that is another alternative that we use, and it is very, very common in head and neck cancer patients who need extractions, and it's definitely something one should consider. Okay, so thank you very much. And before I close my presentation, I would just, as you know, my generation is Instagram generation. I read this quote on Instagram today that don't stop. One day you will be someone's hope, someone's hero. So keep pushing the boundaries. Thank you so much for your time.
Thank you, Anu. I think it's fair to say that anyone that's a clinician in here or a future clinician will definitely see patients who have received definitive or adjuvant radiation, and this is an important skill to develop over your career. Next, we have Karan Patel. All right, and the presentation is osteoclast-mediated cell death in chondrocytes.
Thank you so much. And how do we use this? We just press the timer? The button?
To go next?
No. To go next, this one, and then this one,
Yeah,
and then the laser is this one?
I don't know if it has those.
Oh, yeah, okay. Okay. All right. Is this mic? Okay. Good morning, everyone. Before I begin, quick show of hands. Has anyone broken their bone before? Couple, okay. Perfect. You're not the only one, because around 6.5 million fractures happen in the United States each year, and some fail to heal adequately. As a result, it affects patients' health as well as their lifestyle. And there's been a huge push in fracture repair research. At the Mercutio Lab, one of our goals is to better understand the biological regulation of bone development as well as fracture repair. Now, quick background of bone development. Previously, it was believed that chondrocytes, cartilage-producing cells, served as template for osteoblasts to deposit bone. And then once the bone has been deposited, the chondrocytes would then undergo apoptosis. However, new research has been showing that chondrocytes have a different cell path as well. They have the ability to not only undergo apoptosis, but directly mature into osteoblasts. Now, why is this important? This is important because it potentially increases new therapeutic opportunities with the use of cartilage grafts, where we could use chondrocytes at the sites of where we want bone development, and then with factors, have them convert into osteoblasts and deposit bone. This is important. This is very important towards new therapeutic treatments towards fracture repair, but not much is known on the factors that cause chondrocytes to undergo apoptosis, or rather go to osteoblasts. But as a result, it is important that we understand both cell fates, osteoblasts as well as apoptosis. And this summer, I chose to focus on apoptosis. What could be causing chondrocytes to undergo apoptosis? And we believe that it is osteoclast. One of the major reasons why we believe this is because we've noticed chondrocyte cell death near areas of osteoclast. The cell death is in bright green, and then the osteoclasts are in bright red after trap stain, and then the bright green again is from tunnel staining. So to test this, we came up with a very simple experimental design. If osteoclasts are causing chondrocyte cell death, we should notice them in a cell culture in comparison to just the chondrocyte cells by themselves. We use ATDC5 cells, which are pre-chondrocytic cell lineages, and osteoclasts that we took from bone marrow of mice. And then we set up a very simple transwell setup as well as a co-culture setup. The co-culture setup allows direct interactions between the osteoclasts and chondrocytes, whereas the transwell separates both of them, but allows soluble factors to permeate through both of them. So if interactions are occurring, it's due to indirect contact. And with that, with both of these setups, we fixated them after a certain amount of time and did tunnel and trap staining to visualize this amount of osteoclasts as well as cell death, followed by checking the RNA levels to measure the apoptosis expression. Thirdly, we also did some time-lapse imaging to visualize the cellular interactions, which allowed us to really see whether or not they're interacting with one another in the co-culture. Right here, we use CellPose as a software where we input an image of the total amount of cells. Each dot right here represents a cell nucleus, and once we put it into CellPose, it tells us the total amount of mass and gives us the total amount of cells, as well as cell death and total amount of osteoclasts, as you can see here. So TRAP allows us to see these osteoclasts in bright red. The DAPI allows us to see the cell nucleuses, like I mentioned earlier. Tunnel shows us the dead cells. And once we take the DAPI and tunnel and we merge them, we can confirm that the dead cells are actually with the corresponding nucleus on this other side right here, and that they're not background. With the data that we collected, we were able to see that there is no, in the preliminary data that we collected, there was no significance, but however, we were trying to do a final trial with more samples, as well as a couple changes that we need to include in the next experiment that we learned from this experimental design. I'll talk about that a little bit more. In addition, we checked the expression of the apoptotic genes. We noticed that there was a significant drop in the co-culture as well as the transwell with the osteoclast in comparison to the control in both STAT1 as well as BCL2. And so it's very interesting that we need to keep in mind for the next experiment that we actually measure both of these to the level that we need to and draw significance from it. For STAT1, it could be both pro-apoptotic as well as anti, depending on the cellular interaction and the cellular environment. So it's tough to tell right now whether or not this is inducing apoptosis or not. Whereas on BCL2, it's actually indicative that because of the low expression of BCL2, that apoptosis might be occurring as a result. In addition, we checked the expression for osteoclast genes. We saw expression only in the co-culture. This was as we expected, as the control does not have osteoclasts in them. In addition, the transwell plates have the osteoclasts separated, so they were not supposed to be expressed, and so this was as expected. In addition, we have some live cell imaging over here. Let me play it, if I can find. Good? Oh, there we go. There we go. So the osteoclasts are in bright red right here, interacting with the chondrocytes in gray, and we can see the chondrocytes essentially became smaller and shrunk until it's going to go bright green, which indicates cell death. And so this was the cellular interactions that we were looking for, confirming that they are directly interacting with one another. Whether or not this could be due to due to osteoclasts causing cell death is kind of hard to tell, just because the cell might be just dying naturally. And so that's the purpose of this experiment, is to really see if we can quantify and be able to see if there's a difference between the two, just a control versus having osteoclasts. In conclusion, let me see. In conclusion, our trial run showed that our experimental design was successful in testing for the apoptotic gene expression, as well as the total cells and the total dead cells, and the live cell imaging. However, we are going to be doing further trials with more samples, as well as a couple changes, like I mentioned earlier, and one being that we want osteoclasts to be isolated earlier and grown to greater numbers, as we did not get the amount that we wanted to test them. And if we have osteoclasts to a greater number, maybe we can notice cell death to another greater detail. So that will definitely help us out. In addition, we did some preliminary in vivo testing using the callus of a fractured tibia of a mice, and to see if osteoclasts react to the same, if we can notice cell death there as well. If we do, it would be interesting comparing the in vitro as well as in vivo data. And yeah, we could ultimately decide whether or not osteoclasts are mediating cell death in chondrocytes. Thank you guys so much. I want to quickly acknowledge Dr. Ralph Marcuccia and Dr. Layla Bagheri for supporting me throughout the entire process, as well as the Summer Research Dental Program. They've been completely supportive throughout this process. And any questions? Yes.
That was great. What happens if you block apoptosis? What happens if you block apoptosis in the chondrocytes?
That's one of the reasons why we're trying to figure out. If we can figure out all the factors that can induce apoptosis and ultimately decide how to block it, we can maybe force the chondrocyte to ultimately go the osteoblast conversion. So this is one of the major things that we're testing out, if osteoclasts play a key role, because we've noticed that they're actually very in similar locations and cell death has been occurring there. If we're able to figure out the specific factor later, then we would be able to block that.
So do mutants that have blocked apoptosis have more bone formation?
Ideally we would get more bone reformation, because if they have multiple cell paths, if we can block apoptosis, we can induce the other pathway. And then, like I mentioned, the cartilage grafts, that could be a viable possibility then. Thank you. Thank you so much.
All right, thank you. Next, we have David Sung. David will be presenting on understanding the role of amelogenin self-assembly domain in tooth enamel formation.
Hi, good morning, everyone. My name is David Sung. I'm a fourth-year DDS PhD student in Dr. Ophir Klein's lab. Today, I'll be sharing with you my presentation, understanding the mechanisms of enamel formation of using a mouse model with amelogenesis defects. So my project aims to understand more about amelogenesis imperfecta. This is a disorder that literally translates to imperfect enamel formation. We know some of the causes, including the mutations that cause amelogenesis imperfecta, but the cellular mechanisms are unclear. In my project, I am using a mutant mouse model with severe enamel defects in hopes of identifying cellular mechanisms that are important for enamel formation. As most of you know, amelogenesis imperfecta is a group of disorders characterized by poorly formed enamel. The significance is that it causes a poor quality of life to patients, including ability to digest food properly. As I mentioned, it can be caused by mutations to many different genes, as you can see in the picture on the right. We know some of the causes, but we don't understand enough about the mechanisms that cause poor enamel formation. And in order to do so, we need to first understand how enamel normally forms. in the literature, we know that ameloblasts are specialized cells that contribute to enamel formation. In this diagram here, you can see the mouse incisor and different stages of ameloblasts undergoing differentiation, where they produce proteins and other factors important for mineralization. One of the roles of ameloblasts is to secrete proteins such as amelogenin and ameloblastin, which form the scaffold for mineralization. However, it's still largely unknown what the cellular processes are that occur and what they do to form enamel. A previous study has identified these self-assembly motifs that are important for enamel formation in the protein ameloblastin. And in this study, the authors generated a mouse model where they disrupted the self-assembly motif and showed that these mice had hypomineralized enamel. And similarly, in this paper, the authors also identified that amelogenin also has self-assembly motifs, as you can see highlighted in the red letters here. And as amelogenin is one of the most abundant secreted proteins during amelogenesis, we were curious about how the self-assembly motif in amelogenin affects enamel formation. So we hypothesized that these self-assembly motifs are important for enamel formation as well. We generated another mouse mutant where the self-assembly motif of amelogenin is disrupted. And you can see in these two images, on the left is a histology image showing that the tissue architecture in this mutant mouse is severely impacted. On the right side is a micro-CT that looks at the enamel properties, with the enamel being the bright white region. It's lost in this mutant mouse model, and there's ectopic mineral formed as well. What was also interesting is that our mutant model showed similarities with other models of amelogenesis imperfecta. For instance, this mutant mouse model of enamelin, as well as with of ACP4. So our immediate question was understanding the cellular mechanisms that contribute to the phenotype that we see in our mutant. We were also curious about whether there's common mechanisms between these mutants as well. To begin with, we wanted to look at understanding whether amelogenesis proceeds normally in our mutant. And so we did an immunofluorescence staining of the protein amelogenin. On the top, you can see that in the control where the expression normally occurs, and then on the bottom images, you see in our mutant that the amelogenin protein is still expressed, but the expression pattern is somewhat different. Similarly, we also wanted to look at transcript expression of ameloblastin RNA transcripts using a technique known as RNA-scope. Again, we see that ameloblastin is expressed in the mutant mouse model as well, but there's a very interesting point here where there's ectopic expression, meaning that ameloblastin transcripts were found in regions that we normally don't see it. And it also appeared to overlap with the regions of ectopic mineral formation that we showed previously in the micro-CTs. So we were able to conclude from these experiments that amelogenesis proceeds in our mutant, but we were still unable to identify the specific cellular mechanism that is responsible for this tissue architecture and enamel difference. So we designed a single-cell RNA experiment in which we can observe all of the RNA expression in each individual cell. And this experiment would allow us to find genes that are more expressed in the mutant mice as compared to the control mice. And hopefully we would be able to identify some candidate genes that are responsible for contributing to this phenotype. We performed a pilot experiment where we were able to successfully sequence dental cells from control and mutant mice. And circled here are the ameloblast population, which we identified using looking at their gene expression patterns. Our next steps following this is to perform another single-cell RNA sequencing experiment in order to obtain enough cells so we can reach statistical significance in our analyses. From that, we'll have candidate genes where we can perform in vivo validation from the experiment, and we can also use genetic mouse models to further validate the role of these genes in the process of enamel formation. So in conclusion, I share with you that the self-assembly motif in amelogenin is important for enamel formation, as we see through this mutant mouse model. We realized that the additional experiments are needed to identify the specific mechanisms responsible for maintaining proper enamel formation. And ultimately, we hope from the study to identify one of the cell processes that contribute to amelogenesis imperfecta. So with that, I'd like to thank my mentor, Dr. Afir Klein, as well as all of the members of the Klein Lab and our collaborators at Northwestern University. I'm very happy to, and I also like to acknowledge my funding sources as well, and I'm happy to take any questions now. Thank you.
I don't think I've asked you this recently. I was wondering if you've been able to look at mosaic females yet for MLX loss of function.
Yeah, thanks for the question. We haven't looked yet. We have some samples that we're processing now, and we're very curious to see how females that have essentially a heterozygote genotype are affected in this mutant, and we're very curious to see whether it's as severe or if less severe than the mutant mice that I'm showing here. Yes.
Between male and female, or did you look at both and then?
Yeah, so the question was whether you see any differences between male and female mice. So I guess is to quickly mention also as well, the amelogenin gene is on the X chromosome, so we're curious about whether there's any differences in male and females. No, we have samples, and some of the images I show here are from both female and male mice, but that's definitely a very important point. Thank you. Okay, thanks.
Okay, next we have Eric Yin. So Eric will be presenting on characterizing P75NTR role on osteocytic chondrogenic function in TMJ using CRISPR-Cas9 with his mentor, Dr. Christine Hong. You can.
Okay. Hello, everyone. I hope you have a good afternoon so far listening to all the presentations. My name is Eric Yin, and I'm currently at D1, and let's get right into it. My project is a continuation of Byron's project from last year in some ways, so if you were here last year, maybe you might be familiar with some of the things that's going on today. So let's get into my introduction. So why should you care about the TMJ? TMJ disorders, often abbreviated as TMJD, affect up to 1 in 3 adults. Those are some estimations. So in this room, most likely a sizable amount of you may suffer from some symptoms of TMJ disorders. Even though TMJ disorders affect a large amount of people, the etiology and development of this disease is not really well understood. So my lab is interested in investigating the role that the gene P75NTR has on TMJ development. So what exactly is P75NTR? Well, P75NTR has a well-documented role in the nervous system and is considered a low-affinity neurotrophin receptor. What does low affinity mean? Well, that means that it has a low specificity for all of these different neurotrophins, but can still bind any of them. This is in contrast to high-affinity receptors like TrkA, TrkB, and TrkC, which can only bind maybe 1 or 2 of the neurotrophins. While P75NTR is known for having a role in the nervous system, research has begun to suggest that P75NTR actually may regulate chondrogenesis, so the development of cartilage cells, which you can see up here, and osteogenesis, the development of bone cells, which you can see down here. So naturally, what's your TMJ made out of? Bone and cartilage. So this leads to the suggestion maybe that P75 could also have a role outside of the nervous system and affect TMJ development. So I have 2 aims in my project. The first is to verify whether P75 does play a role in the TMJ, specifically in cartilage and bone development. And secondly, we want to characterize those molecular mechanisms, and we can do that by using CRISPR-Cas9. And finding out those mechanisms means maybe we can find therapeutic targets or ways to treat TMJ disorders. So on to my methods. This is an example of what your condyle in a mouse might look like. You will have your articular disc above and then your condylar cartilage right below it. And these are both going to be chondrocytes or cartilage cells. And first, we want to characterize the role that P75NTR has in the TMJ. So to do that, we can extract the RNA from these cells and send it to a lab for RNA sequencing, which is basically a way of determining gene expression. This is an example of the data that they might return back. We can see in our wild type, so let me go through this slowly because it might be a lot. So you have your wild type samples on the top, and you're going to have your knockout samples on the bottom. So these are samples that have P75, and these are going to be samples that don't. Purple represents upregulated genes, and yellow is going to represent downregulated genes. And each column is going to be an individual gene. So you can see that in our top left corner, all of these genes, all of these ones that I'm kind of circling here, are going to be upregulated when P75 is present. And when P75 isn't present, these same genes are going to be downregulated. This suggests that P75 is responsible for regulating these genes. But what do they actually do? For all we know, these genes could be involved in things that have nothing to do with TMJ development. So we can do a pathway analysis, which sorts these genes into categories listed here. The categories listed in red are related to bone, cartilage, or ECM development. This suggests that all those genes that we just saw were affected by P75NTR actually are going to regulate bone, cartilage, and the ECM. And what does that sound like? To me, it sounds like TMJ development. So keeping this in mind, P75 is normally found in the nervous system and has a role in cell survival, and now we're seeing that it actually might also have a characterized role in chondrogenesis and osteogenesis. So pretty cool stuff that we're finding out here. So remember, P75NTR is a receptor, so now we want to figure out what ligand is actually causing this mediated action. To do that, we can harvest bone marrow-derived mesenchymal stem cells from the femur and tibia of our mouse. You can see the bone marrow cells here. And we can induce chondrogenesis in the presence of various receptors, such as NGF, nerve growth factor, and BDNF, brain-derived neurotrophic factor. And we can stain with Alcium blue to quantify the effect. And I'll talk more about that on this next slide. Okay, so after we induce chondrogenesis, we can stain with Alcium blue, where the dark blue spots are going to represent cartilage. And what we can see is in the presence of BDNF, we're going to have the most amount of area on the well that's going to have cartilage. So effectively, this suggests that BDNF is possibly the main ligand that affects chondrogenesis and is also the main ligand that binds to P75NTR, our neurotrophin receptor. So now that we verified P75NTR is relevant for cartilage and bone development in the TMJ, we want to characterize those molecular mechanisms. And we can do that by generating P75NTR knockouts. And you may be wondering, Eric, didn't you just use knockout cells for your RNA-seq data? Well, yes, I did do that. But the reason why we have to generate knockouts using CRISPR is because in our condyle of our knockout mice, this cartilage is going to be very, very thin. So it's going to be really difficult to harvest the amount of cells necessary to actually to actually assess the signaling pathway of P75NTR. So instead, we're going to generate knockouts with CRISPR-Cas9 to verify our RNA-seq data and also to investigate these molecular mechanisms. This is just an example of a trial run that we've been working on currently of knockout cells on the left. So this is under GFP fluorescence, and you can see there's very few cells that actually are fluorescing GFP, which indicates they've successfully been knocked out. And on the right, you can see all these cells that did not pick up our knockout. So this suggests that our transfection efficiency is really low. We're currently still in the process of working through some of these issues, and that's kind of where we're at in the project right now. So in conclusion, what are the actual takeaways from this study? So far, we found that our RNA-seq data suggests that P75NTR plays a significant role in bone and cartilage formation, and that BDNF is the main ligand responsible. This is backed up by our Alcium blue data, which also suggests that BDNF is the main ligand for P75NTR in forming cartilage. Future steps, we're currently in the process of generating our knockout lines, so our CRISPR knockout lines in various cells. And once we have those knockouts, we plan to characterize the pathways. And if you characterize the pathways, you might be able to find therapeutic targets that could treat TMJ disorders. And hopefully we have some future data to come. So if you're here next year, maybe we'll have something to show for that. And I just want to take a moment to thank my mentors. Obviously, I want to thank Dr. Christine Hong and also my postdoc, Dr. Chiho Kadota, who helped me a lot with my project, as well as some of the dental students here. Byron, thank you for helping me out a lot. And some lab volunteers who are also here, Jonathan and Peter in the back. Thank you guys for helping with my project as well. I also want to thank the Alliston lab for providing us ATDC5 cells, MLOY4 cells, which we use in our CRISPR knockouts, and also Roger Mraz and Dr. Lisa Behrens of the UCSF Research Fellowship Program for helping, do everything here. So thank you. These are my references. Any questions?
Hey, yeah, really nice talk. I just wanted to ask about tying it back to the neuronal component. Do you know if nerves are important at all in the cartilage formation? Yeah, if the NGF could be being released by nerves into that area to help with the cartilage during development?
So P75 NTR is actually like a cell surface receptor. So these ligands are going to bind to it. And what we're theorizing, what we saw in our RNA-seq data is that when the ligand binds to P75, it's going to affect these genes in some way. So when we knock out P75, all these genes are downregulated. So that means P75 must be the one responsible for like actually signaling for it. So there might be a neuronal component to it, but what the genes that P75 normally regulate are related to like cell survival and like the nervous system. But what we found is the genes that P75 is regulating in chondrocytes is actually related to bone development and cartilage development and like ECM formation. And that's kind of interesting.
Thanks.
That was great. So I'm wondering if there are BDNF mouse knockouts or NGF mouse knockouts that can tell you anything about their role in TMJ formation.
Unfortunately, I'm not super certain about that. I know these are just like proteins that bind, so I'm not certain if there is like a BDNF knockout that we can look into, but that's something I appreciate the response, and I'll look into that for sure.
Nice job.
So I will echo that. Very nice presentation and nice work this summer. I'm wondering, when you showed the plot of the upregulation and the downregulation, I noticed that the wild-type mice seem to have a lot more variability. So like wild-type 1 versus the other two. Do you have any recommendations based on that? Do you think it would be helpful to have more wild-type mice and fewer knockout mice in experiments?
Yeah, so the way that we actually got the RNA from our TMJ is we just digested the whole condyle. So there's a possibility that there might be some tissue contamination that may be leading to different, like it's not just all chondral or cartilage cells. It might have like some tissue or like muscle tissue that might be, causing these differences. And that's part of why we want to create these CRISPR knockouts, is to verify like, is this data actually accurate or not? Because there's a possibility that this is not entirely like all cartilage cells, you know what I mean? And that's also what I was saying with the knockout cells. The cartilage is very, very thin, so we're just assuming that the ones that we took were the actual cartilage cells, but that's an assumption, and we want to verify that. So that's why we really need to generate these knockouts in vitro, and we can't just assume that our knockout data from this RNA-seq is 100% like factual. It must be this way. So thank you for the question. No.
It'll be easy. Yeah, really great talk. I was just curious for the sequencing data, is this all males or females, or is this kind of
Yeah, this is all male. So this is male versus knockout male, yeah. Thank you.
All right, let's give one last round of applause for all of our presenters this morning. So I'm excited to introduce our next speaker, Dr. Piri Velupillai. Dr. Velupillai is a dentist and an oral medicine specialist specializing in inflammatory diseases and chronic conditions of the mouth, including precancerous changes and oral manifestations of HIV/AIDS. She earned her DDS from the Karolinska Institute in Sweden and her master's in oral biology while completing her oral medicine residency here at UCSF. She's a member of the American Academy of Oral Medicine and the American Board of Oral Medicine. She's been on faculty since 2003, has an active clinical practice, and has trained many students and residents, including myself when I was at GPR here. So I'm very excited to introduce her. She's going to be speaking on the use of topicals in oral medicine. So please help me in welcoming Dr. Velupillai.
Thank you, Dr. Lugo, for that nice introduction. And I am not sure about you, but I am starting to get very hungry, so I'll try to keep it quick, the presentation. So, let's see. So it's just a boring slide here. So because I hear that most of you are first and second year dental students here, so probably don't have much of an experience with oral medicine specialists. So I wanted to give you a little introduction. So when I say I'm an oral medicine specialist, I get people ask me, what is oral medicine and what exactly do you do? So these are the things I put up the slide. You can read it at your own time and understand what is oral medicine. So to make it easier to understand, I tell them it is like a dermatologist, a dermatologist treating skin diseases. We treat oral mucosal disease in the mouth. So it's a dermatologist in the mouth kind of question. And then we also get questions like, oh, so if you diagnose diseases like oral pathologists do, so what's the difference between oral pathologists and oral medicine specialists? So I get that question a lot. So we are the clinical side of oral pathologists. We interact with patients. We collect data and do biopsies and send the biopsies to the pathologist, who will look it under the microscope and then give a diagnosis, which we then bring it back to the patient and then treat the patient accordingly. So that's our role in oral medicine. So one of the things that the most important thing that we do or end up doing in oral medicine is prescribing medications, as my residents and third-year rotating dental students will know when they rotate a week with us. So let me just give you a quick review on the medications. So the most frequently prescribed medications are immunosuppressive medications, pain medications, and then, of course, the antifungals, antibiotics, and antivirals. So since we treat a lot of autoimmune conditions, inflammatory diseases, we work a lot with immunosuppressive medications such as corticosteroids and prednisone. So we're moving on to topicals, which is my topic today, topical medications. So there are rinses, and let me go back to it, gels, ointments, pastes, and lozenges, and powder. So it's nystatin powder, especially when you get fungal infection under the denture. So we use the nystatin powder to treat the fungal infection under the denture. So the part that I'm going to talk about, there are two parts to my presentation. One is how the topicals work alongside the systemic medications when we treat patients. And the next part is a new emerging topical and how we are planning to bring that into practice. So the first part, to illustrate the first part, I want to give you an example of a case that I still see, still continue to follow up since 2021. So this is a 52-year-old male, was sent to us by his medical team for recurrent and painful lesions with candidiasis. Candidiasis is a fungal infection. So the patient's chief complaint is pain inside his cheeks, difficulty swallowing, and some with hoarseness that he was having difficulty with. So the medical community has already diagnosed this patient with fungal infection in the mouth, and they have prescribed multiple times, and the patient will partially respond, and then when they stop the treatment, we'll get the full-blown fungal infection. So to understand this case, before I start showing you pictures, I want to briefly talk about his medical history. So the patient was, a year before he came to us, he was diagnosed with myasthenia gravis. It's an autoimmune neuromuscular disease where the muscle weakness takes over. So this was believed to be, when he was diagnosed with myasthenia gravis, they found out that he has a tumor in the thymus gland, which they believed then the thymus tumor was driving this myasthenia gravis condition. So they tried to treat it by removing the thymus gland by doing radiation, trying to get it to control the myasthenia gravis. he seemed to get better, but the myasthenia gravis, he started showing symptoms of myasthenia gravis again. So they investigated further and found out that the tumor had actually metastasized. Therefore, he was starting to get myasthenia gravis symptoms again. And in the meantime, while they were trying to treat the tumor, the metastatic disease by radiation, resection, and chemotherapy, and even rituximab immunotherapy, he started getting immunodeficient, and that brought on so many problems. So that's how he ended up with us, with recurrent, difficult to treat fungal infection. Okay, so this is where I feel comfortable with. So as you can see, the patient's mouth, he comes with severe inflammation with white plaques and yellow, whitish yellow plaques, and lots of ulcerations, small cuts, and swelling. Anyway, let me show this. Which is the light? Oh, the light. I was going to the light.
Oh, it's this one.
Okay. Okay. So in here we see a large ulceration, and there were little slits-like ulcerations and linear ulcerations on the buccal mucosa, which was causing him pain. He was more worried about his hoarseness and difficulty swallowing more than anything else happening in the mouth. But of course, one of the complaints was difficulty eating. So as the medical team also had asked, we decided to do a biopsy from his cheek, which turned out to be, of course, as expected. There was candidiasis, as you can see in the picture, and also it came out as lichen planus, chronic inflammatory disease that on top of the candidiasis that he had mouth, he had lichen planus, which was the reason why he was getting all those ulcers. So, as usual, when patients come with these conditions, we start treating them with topical medications. So that was the initial treatment, started with the dexamethasone, a rinse, and a gel fluocinamide. These are all corticosteroids. And then also the fungal infection, we started treating with another topical antifungal this time, a clotrimazole, a full treatment. And we were hoping that he would find some relief. But he was not unsurprising. So he wasn't finding much relief. Therefore, we then upped our game to prescribe systemic medications, and this time antisystemic, antifungal, increased dose, and then also prednisone because his ulcerations weren't healing. So to treat the underlying lichen planus, so we combined the prednisone, high dose of prednisone, with antifungals and treated him with this combination, and also kept the topicals. Some part of his symptoms improved, the hoarseness and the swallowing improved, but he was starting to get a lot of pain from the tongue. So when we could see, as we saw in the initial pictures, there was a ulcer on the tongue dorsum, which then we decided to biopsy. So the biopsy, I'm glad we biopsied because we thought this also was part of the lichen planus, but the biopsy then went to show us that there is HSV, herpes simplex virus, in the ulceration, which prevented it from healing, prevented or made it worse. Why? Because the patient not only was immunosuppressed, we started with the prednisone, high dose of prednisone, which made him further immunosuppressed and making him susceptible to viral infection, which caused this deep ulcer on his tongue. So this was, again, led us to treat him with high doses of antiviral for 7 days, and then so the prednisone, and we understood the prednisone, high dose of prednisone may have triggered this, so also try to taper the prednisone alongside it, and then continue to treat the patient with topicals. The patient started to feel better. And we thought our job was done. We have made him feel better, and this is it. He can maintain his status from now on. The patient then, 2 months later, we found out the patient was hospitalized. He returned to us with similar complaints, not as bad as the first time, similar complaints with new development in his medical history. So we found out that he had neutropenic fever. Sorry. He had neutropenic fever, and then he also, his B cells continued to drop. So they held off his immunosuppressive therapy with rituximab, which was for the tumor, and prednisone. All the immunosuppressive therapy was taken off, and he still didn't improve. His immunoglobulins kept dropping, and therefore they then diagnosed him with a new diagnosis on top of the diagnosis he already had with hypogammaglobulinemia. It's very difficult to say the entire word. This basically means that there's low B cells, and therefore he's immunodeficient. So with this new immunodeficiency, hypogammaglobulinemia, and the thymoma, which in his condition, it's a metastatic thymus cancer, and the myasthenia gravis, all in this together, is called the Good syndrome, a phenomenon that's been described. It's a very rare condition, but it's been described in the literature, and this is why we finally figured out it is. This is why the patient was so hard to treat. So, again, with the medical community now understanding why this patient was hard to treat and why this his immune status deteriorating. So now the treatment were then adjusted. So they were going after the metastatic disease in his lungs, the thymus metastatic disease in the lungs, to treat it so that he can his symptoms can improve. So far, they haven't been able to improve his systemic disease, but his oral conditions have improved. So again, it doesn't stay like this. He does get flare-ups of lichen planus on and off, and he continues to come to our clinic every month, even now to this day. Sometimes with new complaints, but we are able to now, knowing the disease behind this, we're able to kind of maintain him, and so that he can continue to live a productive life, still working full time, and preserve the quality of life that he wants to with his family. So that was my first part of the talk. I think I went too much. The second part, this is something that I've been thinking about lately, a new topical that's been in our minds. I wanted 3 different cases quickly, pictures of 3 different cases. Let me quickly go to the pictures because this is otherwise healthy 44-year-old female with white lesion for 2 years, had been diagnosed mild dysplasia once, but clinically a leukoplakic patient. So we'll go on to the next one. I'm going to rush through this so I can get to the pictures. This is the second case, 69-year-old female with, again, asymptomatic white lesion, this time on the tongue. And multiple biopsies during the course of this 5 years showed dysplasia and sometimes candidiasis. We would treat the candidiasis, but the lesion will still remain. So that then we call this leukoplakia. And the third patient also, that's the second patient, and then the third patient is 83-year-old female, also with a white lesion on the tongue. And the last biopsy showed mild dysplasia. But she has a significant history here because she had squamous cell carcinoma on the left side of the tongue. So with this in mind, all these 3 patients were asked to go get a surgical opinion about removal of leukoplakias. But all 3 of them were hesitant for the surgical option, wanted to know if there's anything else they can do. In the meantime, if the surgery becomes necessary, they will go on to get the surgery, but they wanted to know if there's any other alternative. So Okay, so that was the third case there. So the patient that I kind of went over quickly. So what is leukoplakia? So again, I don't have to spend too much time. Here, it's a well-demarcated white plaque in the oral mucosa lesion that is not attributed to any other cause. And it's estimated in the population about 5 to 4.5%. How do we manage leukoplakia? So after incisional biopsy, we always give the patient of surgical options, scalpel removal or laser excision or laser ablation to photodynamic therapy. And otherwise, if patient chooses not to go through surgery for some reason, or if they didn't want to, then they would be followed up with incisional biopsy, close following up with incisional biopsies. There's systemic and topical treatment that has been used and studied, retinoids and herbal extracts and bleomycin, not with so much success, again, not used as much anymore. So now there's 2 new topicals that have come up again. Imiquimod, it's an immunomodulatory agent, and of course 5-FU, a chemotherapy agent used for cancer chemotherapy, but used as topicals for leukoplakias and dysplasias in the mouth. So we got interested, or at least I got interested in imiquimod when I talked to my colleagues at Harvard, who also happened to start using this more in their patients with leukoplakia. So in oral medicine and literature that coming from oral medicine, there is not much evidence out there to treat with imiquimod in oral leukoplakia. But in dermatology, there is a plethora of evidence for conditions like anogenital warts and actinic keratosis. But in oral medicine, we used to use this imiquimod for intraoral warts when we saw intraoral warts in HIV patients a lot in oral medicine clinic in the early 2000s. We would we would prescribe this medication to treat these patients. Since we don't see the warts or the HIV patients back in our clinic, since the antiretrovirals improved, we have stopped using that. But now, since the use in leukoplakia precancerous lesions have become a new idea, so we wanted to kind of try it in some of these patients who didn't want to go through their surgical option. So imiquimod is basically a immunomodulatory agent. It's a cream. It's formulated in a cream. In a cream formulation, and it has good skin penetration. So this is all from the derm literature. The mechanism is explained as antiviral, antitumoral effect. It is a known toll-like receptor 7 agonist, and through which the cell-mediated response, which will then help the dysplastic cells to work on the dysplastic cells. So we decided to use this. Again, it's not approved for dysplasia or oral dysplasia, oral lesions, but we decided to use that in our patients with educating our patients of the risks, and with the experience and with the data from the derm literature, we decided to use it in our patients. So this was the case 1 when I showed you, one of the 3 cases, the first one. So she was asked to apply. So the one course of application would be once every day for 5 days and 2 weeks, 2 days off, 5 days on, 2 weekdays off for 30 days. So I'm sorry here. So we see how gradually the lesion throughout the treatment disappeared completely. This was one of the successful cases. You know, not every case turned out to be like this, but I just want to show you we were excited to see, we were very happy to see the response. The patient did have some side effects, but she was, when she completed her course, all the side effects went away, the symptoms, the local irritation, everything went away, and this was, the last picture was 3 months post-op, and it completely cleared the leukoplakia. The second case is the leukoplakia on the lateral tongue, which we saw the patient who is completely asymptomatic would have fungal infections on and off. So she used it on her tongue. She had a little more side effects and complaints. And this is after the first treatment completion, the picture there. And we would see the reduction. This is the leukoplakia, the demarcated plaque-like area. So that kind of reduced into 3 little small areas of leukoplakia there. So she had some response, but not complete. The third case is the patient, 83-year-old woman with history of squamous cell carcinoma on the other side of the tongue. When she applied the first 3 courses, 3 30-day courses, she didn't have any side effects, no change in the lesion. She was surprised that she didn't have it. She was worried. She kept coming, is this working? Am I doing the right thing? So after the third round of treatment, I'm just pushing the wrong button here again. Third round of treatment, we saw some reduction of the lesion. So again, no side effects, no irritation. She just started slowly responding. And by the end of the 4th course of the treatment, it had dramatically reduced. But again, remember, some patients responded with the first try. This is the 4th try, and the lesion started decreasing in size. So we were again encouraged to use this, but we will carefully keep monitoring the patients, because especially this patient who has had cancer on the other side. So we will biopsy when she returns from her hip surgery. She's having a hip surgery right now, and biopsy this lesion to see if there is any disease activity or any progression of dysplasia in this case. So when we started using this, of course, like I mentioned, we 2 other colleagues at Harvard also used this agent, treating their own patients as well. So instead of publishing, we wanted to look at the retrospective data and publish to see, because in order to plan a clinical trial or any trials after this to start producing data, we wanted to have some kind of pilot data or some kind of retrospective idea to how to move forward. So the Harvard group also wanted to do that, so we decided to combine the data together to have more impact from this retrospective data analysis. So the abstract was presented at the AAO meeting with the American Academy of Oral Medicine in May 2023, and there's a publication which was submitted but not published yet. So under the title of Imicmod for the treatment of oral leukoplakia, a 2-center retrospective study, the Harvard and UCSF combined. So, Harvard also used this with the gingival trays, custom-made trays to keep the medicine in place, because it's a cream, it slides off easily. We asked our patients to use a piece of gauze to keep the medication in, and but in the Harvard group, they used the trays, which helped also keep the absorption minimal outside the lesion. Okay, so we 100% resolution. So there are total number of patients here were 38, and 100% resolution was seen in 42% of this case number of cases. So I think in the interest of time, I'm going to quickly go through the next slide of adverse effects, because this is, I think, very important, since there is still some trepidation among oral medicine community and surgical community with use of this medication, because it's an immunomodulatory medication, what it can do to other things, what other immune problems it can trigger while using this medication. Even though it has proven to be safe in derm literature, and even though it doesn't have too many side effects, it is still something hotly debated among ourselves, whether this is something we want to continue with. I think with more data, the use of this imiquimod in oral cavity will help us understand more. More studies will give us information to use. This is some more data from the Harvard group. They have a little better lower gingiva pictures here. So I wanted to share that with you. You can see how the pre-treatment lesions and during the treatment, how the lesions tend to become more chalky in appearance and red surrounding areas, which is inflammation because it causes immune reaction around it, and then slowly starting to disappear and then stays disappeared. So that's the promising part of this. Instead of a surgery that might cause more tissue loss, this is, I think, very exciting that we can see this helping this, precancerous patients. Okay, so again, to just emphasize, 68% of the patients in both groups showed more than 50% improvement, 68% of the patients. So that itself is, I think, motivation for us to continue studying this more and give it, to use it in a different ways to find a different formulation so that it stays better in the mouth rather than slip to go to other areas, so that it can be used efficiently. So what are the side effects? Except for local irritation, it was well tolerated. There were, of course, some systemic side effects were reported, flu-like symptoms, fatigue, loss of appetite, but those all resolved when the patient stopped the treatment. During the treatment, if patients experience more side effects, we would have them take a break from the treatment, and when the patients start feeling better, we would have them start again. So that's one of the new topicals I was feeling very excited about, which I wanted to share with you. The first and second year oral dental students, which I'm happy to see them when they come for rotations in their third year. So, of course, we know that more research need to be done, whether this could be treatment alone or whether this can be combined with surgery or laser treatment, which is, again, I think there's a lot of potential to this. And with this, I wanted to just conclude my talk and let you ask questions, because I feel better when, rather than presenting, I feel better when questions come. Thank you.
Thank you so much, Dr. Villeda. So I wanted to extend our gratitude to you for presenting your lecture today and present this plaque, the only kind of dental plaque you want to have. Maybe you can stand over here. I think there's a photo. But I think that in the interest of time, we have to cut you off and cut off questions. So if you have questions, please come and talk to our presenter and all of our other presenters. I want to thank Dr. Lugo for being an excellent moderator. I want to remind people that there are post-lunch across the street and posters. So please go see all the student posters and come back here at 1:30 for the next set of talks.